Now I have to repeat the same thing. Yeah, I'll start again. Now, ready? All right. So, hello, good morning, everyone. My name is Mahesha Manjunatha Poojary, and welcome to the day 3 of the Gian program. So, the next generation sustainable alternative proteins for food fundamentals and technological innovations. And today we're going to have two important lectures. Both are related to analytical chemistry. The first one is how can we use chromatography and spectroscopy to characterize alternative proteins or proteins in general. And the second topic is specific about how can we use chromatography and mass spectrometry to characterize proteins. So, that will be a total of 3 hours lectures for today. All right, so I hope you have had an amazing yesterday. So, I really want to have this particular question for you. Did you explore Rourkela? No, not yet. So, it's no time. Good. Maybe the second question, the most important one. What was the most interesting thing you learned yesterday? So, yesterday we were discussing about market opportunities and so on, and then we went deep into different extraction technologies, conventional extraction and emerging technologies to extract proteins from different biomasses, right? So, what was the most interesting thing you learned? Could anybody raise your hand and start something, mention something? Nothing was interesting? Okay.
Non-conventional methods of extraction.
That was interesting, and why do you think so?
Principles are different from one another.
Yes.
Ultrasonication is different, enzyme assisted is different, and they have a particular substrate to work on. Every time we have to optimize, which is not generalized like alkali extraction.
Exactly.
Or like salting out, salting in, which is generalized, but here it is specific to proteins.
Absolutely, yeah. So, what she is telling that this technology is really good, however, they have their own advantages and limitations. You need to optimize perhaps samples and processing conditions to get good yield or good purity and so on. Do you have any point, Asta? Yeah.
I was really amazed to know about the, like you said that you had personally experienced the this alternative plant market in the USA, and we do have a market of gluten-free products in our India, which is growing on. But certainly in the coming years also we'll have this market trend of alternative proteins in the supermarket of India.
Yes, absolutely.
So I was really amazed that there were so many varieties of the products which have been launched in the market, and they are being used by the consumer.
Yes. So India is a big one of the biggest markets because of our population. So perhaps I would say that India is an emerging market for alternative proteins. Maybe in next 5-10 years we can see more varieties of products in the supermarket shelves. Yeah.
So, you showed slides about your visit to that market, something products. That is quite interesting for me.
That's cool.
We cannot go there. You
yes.
bringing us.
Yes. That was a good experience for sure. Yes. Good. Any other comments? No. Okay, that's good to hear, and let's move on to today's lecture then, and I'm going to start with the analytical techniques for protein characterization, chromatography and spectroscopy. There's a small change. I'll start with spectroscopy first and then I'll go to chromatography, because then my flow of teaching will be nice according to me, because spectroscopy is complicated technique, but it's much simpler compared to chromatography to explain as a teacher. So, I'll start with the spectroscopy and then I'll go with chromatography. Is that okay with you? Excellent. So, how many of you have used any spectroscopic methods to analyze or characterize proteins? Only one person? Never used any spectroscopic technique? You have used? I can give some examples. Infrared spectroscopy, Raman spectroscopy, UV spectroscopy, NIR spectroscopy, NMR, IR, there are many spectroscopic techniques today to characterize proteins. Can you recall one or more that you have used?
And NMR I use, sir.
IR and NMR, yes, these are the most common techniques that we use to characterize many of the biomolecules. Excellent. So, I am not going to cover every spectroscopic technique, but I am going to cover some of the most important things that we use today. Okay. So, my first question is, why do we have to characterize proteins? Before coming to this question, I'll try to link my today's lecture with yesterday's lecture. Okay. So, yesterday remember that we had let us say a microalgae, a cell containing proteins inside it, right. Then we also have a plant cell containing proteins inside it, and then we also have a seed containing protein inside it, right. Then we discussed a lot about, okay, it is very important to extract proteins from them, right. And for extraction we have discussed a lot of different techniques, conventional and non-conventional technique. We talked about salt extraction, we talked about alkali extraction, we talked about enzyme assisted extraction, ultrasound assisted extraction. I did not cover microwave assisted extraction, I also did not cover high pressure homogenization based extraction. We also covered about pulsed electric field extraction. There is a lot of extraction techniques we discussed. Okay. After extracting we get something, right. Usually we get two fractions, just to make it more simpler way, we have a supernatant and we have a pellet, okay, after doing some XYZ treatments. And in both the fractions we might have some proteins, right. Then we discussed little bit about, okay, this can be subfractionated and so on, and at the end of the day we get a powder, right, we get a powder, and this powder, let us say you do freeze drying or you do spray drying or you do vacuum drying, I do not care, you get at the end of the day powder, and this powder contains different percentage of proteins. Do you agree? So, maybe 50% protein, maybe 75% protein and 90% protein, right. And I also mentioned to you that depending on the percentage of these proteins, they find different applications, right. Some will go to bread making, some will go to let us say breakfast cereal making for example, and some will go to maybe infant formula making or meat analog making or plant based milk making. So, different applications we have seen, right. But one of the important thing I did not cover yesterday was, we all discussed a lot about amount, purity, we discussed that one, yield versus purity and those things we discussed. But another important thing is about the quality of the protein, right. And the quality is something related to the structure of the protein and the composition of the proteins basically, right. And that determines at the end of the day functionality of this protein. Do you agree? Functionality, what we talk techno-functional behavior, for example, water holding capacity, oil holding capacity, gelation, emulsification, viscosity, and all these things at the end of the day they are related to the structure of the protein, right. Because how the protein is behaving in solution or in solid state determines the structure, texture, flavor of food products. So, structure is very important, remember. And structure is also very important because protein in food system does not stay alone. Do you agree? We never drink a solution containing only proteins. It is very rare in the rarest case, right. It is bland, no taste, and I do not think people will happy to drink just protein powder like this one. They are always added with something else. Most of the cases you have sugars, carbohydrates are added, minerals are added, other emulsifiers are added, polyphenols are added, many things are added to get the final food product. Do you agree? Now, the protein is never alone in the food products, they are always trying to interact with some other biomolecules. Do you agree? So, protein is interacting with some other molecules which will be carbohydrates, it could be minerals, it could be lipids, anything. So, in the complex food system there is a lot of interaction. Please note that the interaction between proteins and other food molecules also determines the functionality of the protein itself. or the taste and flavor or texture of the final product. Do you agree? So protein is an important component. It's never alone in food system. It behaves differently depending on with whom it is interacting with in a given food system. Do you agree? And all this interaction is also dependent on the structure of the protein. Okay? Some proteins can interact better with carbohydrate and some cannot. It's simply because their amino acid composition, their folding behavior could be different, so they can interact with proteins in a different level. Okay? Now, I'm again and again using the word the structure, right? The structure and the composition of the protein is very important. Now, let me come back to a little bit about the composition. What do you mean by the composition of the protein? It's a very generic term. We can do it in two ways. When in my very first class on day 1, I mentioned about proteins are made up of what? Amino acids. We talk about protein composition could be amino acid composition, right? Proteins will be having a variable content of different amino acid then, and that changes the structure of the protein, and that also changes the solubility of the protein, that also changes the nutritional value of the proteins, that we have already known. However, in addition to this, maybe I should have not written here because all right. In addition to the individual composition of an individual protein, let's take a plant protein as an example. Now I get a pea protein isolate. This is pea protein isolate, okay? And I put it into water like this. And then it is dispersed in the water or dissolved in water, I don't care. Now, whether this pea protein isolate contains one protein or multiple proteins. Multiple types of protein. Please note that one. Plant proteins are extremely complex. When I say pea protein isolate, when I say pea protein consolate, when I say soy protein isolate, when I say rapeseed protein isolate, they are not a single protein. They contain too many different types of proteins. In my very first class, we discussed about, okay, we have globulins, we have albumins, and we have prolamines and so other things, okay? And globulins are also not a single type of protein. There is a lot of things, convacillin, glycellin and all these things, right? Then again they are subdivided into different classes, right? Now I write globulin 1 and globulin 2. And that is not the end of the story, because each type of globulin or each type of albumin again might contain different types of isoforms of the protein. What do you mean by isoforms? Imagine I have let's say rapeseed protein contain a protein called napin. Okay, one of the protein in rapeseed is called nepin. Okay, nepin is an I think it's an albumin basically, but when I say nepin, it's not an individual protein. It's basically nepins plural in seed. Why? Because the amino acid composition in nepin could be slightly different in different types of nepins. So there is isoformic abundance will be different. Now you see that how plant proteins are highly complicated system compared to say dairy proteins, right? You have a isolate, but the isolate is not an individual protein, and they are divided into albumins and globulins and prolamines in a different proportions, and each proteins might have their own subvarieties, isoforms, proteoforms and so on. So at the end of the day, it's a complex mixture of different types of proteins. Now, all these different types of protein and their proportion affects the functionality. Make sense? When I say, for example, you are working with let's say pea proteins, and you say that pea protein isolate is having very high water holding capacity. Remember one thing, it's not only one protein is responsible for that increased water holding capacity, it's actually multiple proteins are responsible for that one. Now, if you dig a little bit deeper into this particular subject, we need to understand what exactly happening in this complex protein matrix. Who is doing the job? Which protein is responsible for increased nutritional value? Which particular isoform is responsible for increased water holding capacity? Which isoform is more soluble in water? Which particular fraction is more soluble in alcohol? And these things is extremely important to get the overall functional properties of a given protein matrix. Does that make sense? And that's all we call it as characterization, right? It's a very big word, characterization. It's basically understanding the structure and composition of proteins. Make sense? I gave a long introduction just to give you an idea that it's an important topic, right? Without understanding the structure and composition, it's very difficult for you to understand functionality changes in a given food system. Okay? All right. I found this figure from this particular website. I really enjoyed it. So basically, when you say protein characterization, we talk more about the structure. Okay? And then we also talk about the molecular weight, because each protein will have its own molecular weight, and the molecular weight is a fundamental aspect of the protein that again determines the solubility, functionality, and so on, right? And the charge, we discussed about charge also in our very first class, positively charged proteins, negatively charged protein, amphoteric proteins are amphoteric, right? We have an isoelectric point, right? And then, as I mentioned in my previous slide, protein is never stays alone in a food system. It will always try to interact with other biomolecule. Other biomolecule could be protein itself. Okay, protein can always interact with another protein. In addition to that, protein can interact with carbohydrates, polyphenols, lipids, and so on. So all this will change at the end of the day, the functional properties of proteins. And we also have something about protein purity and post-translational modification. That means especially when plants generate proteins and they never simply exist as an individual beautiful proteins like an amino acids, as I showed in my previous slide, one of the amino acids may be modified. There may be phosphorylation, there may be glycosylation. So amino acids might be modified to a lesser or greater extent, right, within the cells itself. And that will again change the functional properties of the protein. Please note this point. So amino acids can be modified within the plant matrix itself before you extract and use it, and that is called post-translational modification. For example, phosphorylation is one of the most common modification. You will add a phosphor moieties to the proteins. Glycosylation, you will add a sugar moiety to the proteins, and that will change the solubility of the protein. And to identify all this modification, we need to start characterizing the proteins using different technologies. Okay? So I'm going to cover some of these technologies today. I must tell you that I'm not going to cover everything because I don't have that much time, but I'll only show you some of the most important ones we are using today. But if you have other techniques, we are welcome to discuss as a discussion. Okay? Now let's move on to the structure. In my very first class, I mentioned to you that proteins are having 4 different structures, all right. Primary structure is simply nothing but its amino acid composition, and the secondary structure is nothing but you have hydrogen bonding. You have hydrogen bonding which will make proteins to coil, protein act as a sheet, we call it as alpha helix and beta sheets, and also there is something called random coils. And in addition to that, proteins can be 3-dimensionally folded. Proteins are never linear in food system or in biological system. They are actually 3-dimensionally folded because of, let's say, disulfide bonds, hydrogen bonding, hydrophobic interactions. So the proteins are bent in a way that it gives a particular shape to the protein, right? And that's called tertiary structure. And tertiary structure is also important for protein-protein interaction, protein-carbohydrate interaction, protein-polyphenol interaction, and so on. Right? And then we have a quaternary structure where different protein subunits can make a dimer, trimer, hexameric structure of very big complexes of protein. So these are the 4 different possibilities or the structural variations a given protein can have. I have told you all these things before only, right? Now I'm going to show you a little bit about an overview of how different techniques can we use to reveal these structures. Can you tell me why the primary structure is important for a given protein? Why primary structure is important? Sorry, yeah.
primary structure contains all the information stored, like genetic information is also stored in primary, and it is a base for next this secondary or tertiary structures.
Absolutely, yeah. So he's telling that it's an important measurement we have to do the primary structure analysis because it also stores the genetic information because proteins are produced based on the translation within the cells, right?
And presence of specific amino acids also defines the functionality of that particular protein, because on the basis of, let's say, cysteine is present, so that is help in the formation of disulfide bond, and that gives the strength in case we talk about the gluten protein.
Absolutely. So the amino acid composition is also very important because that will talk about the structure and also the, for example, how disulfide bonds can be formed within the primary structure, and that talks about again stretchability of the matrix, the Dow here, and that's in other words is the functionality, right? She has a point there.
The primary sequence that helps, that determines how the secondary structure, tertiary structure and quaternary structure will be formed. So that plays a major role, how based on the primary sequence, how it gets folded to form the next structure, and that determines the
Tertiary
3D
structure.
structure as well.
Excellent. So what she is telling that the primary structure is the fundamental, the basis of the proteins. Basically, based on the primary structure, then the secondary structure takes place, and then tertiary, and then the quaternary, because the amino acid composition and the sequence of the amino acid is very important to determine the final tertiary or the quaternary structure of the protein. Fully agree with her. Now we mentioned about 2 points here. One is composition. Composition is nothing but how much lysine we have, how much glycines we have, how much tryptophan we have. But the protein is not just having how many of these amino acids. It's also important to learn how they are oriented, how they are in a sequence, like whether we have a glycine after lysine and tryptophan, or we have a different combination. Okay, I have tryptophan, lysine and glycine. Now these two, let's say tripeptides, are very different. Do you agree with me? A sequence is also very important because based on the sequence, the pI of the protein can vary, because the neighboring amino acids will determine how the other amino acids behave. So that's the reason why the sequence is also important. In addition to pI, the sequence also determines how protein can bend, like happens because of let's say 2 thiol group. When they join together, you get a disulfide bond like this one, right? If they are far away, if the protein cannot bend, the disulfide bond never happen, right? If they are too near, then you still have a very steric strain, they cannot react together. They should be present in a particular distance. The protein should be able to bend. The bending is also dependent on which amino acid is present in the backbone. Make sense? And that's the reason why how individual amino acids are located one after another, the sequence, that's also very important, right? And that determines the secondary, tertiary and the quaternary structure. So determine the primary structure, the first thing we do the amino acid composition analysis, often done by HPLC or GC. Then we do, to find out the sequence and also the composition, we're going to use Eadman degradation, right? And then we can also use mass spectrometry to determine the sequence, but it's a little bit difficult, especially if you have a very big protein. If the polypeptide of like 20, 30 amino acids, you can easily find out sequence. But if it's a huge proteins with like 3,000, no, not 3,000, 300 to 400 amino acids, it becomes a little bit tricky. So often we use Eadman's degradation to find out the protein sequence. I'll get back to that later. Then we have a protein secondary structure. Remember, it is I have only shown the alpha helix here. We also have a beta sheet. And the secondary structure can be determined by FTIR and CD. CD is circular dichroism spectroscopy. And the tertiary structure, we can use fluorescence spectroscopy, X-ray, NMR, and also electron microscopy. Okay? And the quaternary structure, you can use dynamic light scattering, size exclusion chromatography, X-ray, NMR, and so on. Please note that one technique will never give you the full information, right? That's a problem or the difficulty I'm mentioning here in protein analysis. It's often combination of the different techniques gives you the overall structural information. Please note that in order to get a structure of one protein, it might be 3 PhD work. I'm telling, it's not easy, right? For example, you have isolated one particular protein from like a new plant-based matrix, and nobody knows what protein is it, right? Now you want to know what is the nature of this protein. Then you will start with analyzing its primary structure. Okay, I know its amino acid composition. And then you will analyze, okay, its secondary structure. Then you will analyze its tertiary structure. And then you will analyze whether it can make a hexameric structure and so on, quaternary structure. And all these things are very, very tedious processes. Of course, we do have now automated system. We do have artificial intelligence to help us in structure determination. However, it's a very, very complicated and tedious processes. In most of our laboratory work, for example, you are a PhD student, you don't do everything. Do you agree with me? You just see, okay, I just want to know whether the change in the secondary structure. I have a FTIR in my lab, I'll use FTIR. Oh, I do have an X-ray, in collaboration. Okay, can I make a single crystal of protein? Yes, now my protein is crystallizing. I'll take an X-ray of that one. Now I get a beautiful 3-dimensional structure of the protein. But again, it's another PhD work, right? It varies from labs to lab, it varies from PhD to PhD, which analytical tool you're using to analyze the structure and the composition of the proteins, right? And if you cannot use all the techniques, don't worry, it's not possible, nobody uses it. Unless you have 10 PhDs working on one protein, you can do that, okay? As I mentioned, I'm not going to cover everything, but I'm covering some of these things. And X-ray is an important tool to measure protein structure. A very, very nice technique, gives almost complete information about protein composition and also. Good. Now I'll talk a little bit about the primary structure determination. Please note that I'm not going in detail about this technique because in food science we are not doing that much. We often never determine primary structure of the protein in food science. I don't say we never, it's because it's a little bit complicated and we don't need that information in most cases, and that's the reason why we don't do it. However, if you look into the biological scientists who are studying about enzymes, they're studying about antibodies and so on, they always try to determine the primary structure. So I just give an example how we determine that one. So you know that protein has amino acids, these are all amino acids, okay? And there is something called N-terminal and there is something called C-terminal, right? And to determine the amino acid sequence, you need to know which is present one after another. Okay, lysine after cysteine and glycine, leucine, isoleucine and so on. We need to find out which one is present one after other, right? To determine that, what they do is they use a reagent called Eadman's reagent, it's called Eadman's degradation, and this reagent interacts with the N-terminal amino acid, the last one. Okay, and then we get a compound called phenyl thiocarbamyl derivative. It looks like this one. It's a fake structure. I'm not going to teach that one, as I said. And then what we do is that then this reaction is done in alkaline condition, and then we heat it, and we make it a little bit acidic, and then this particular group will be cleaved off here, and then we get an individual unit. Okay, now it's connected to an amino acid. Can you see? This is an amino acid, and we have a derivative. And this can be analyzed by, let's say, HPLC, right? So now I know, let's say I don't know what is the S amino acid, I don't remember the proteomics, serine, okay. Now let's say it is serine. Now we came to know that the N-terminal, the first amino acid is serine, okay. Then we lost this one, do you agree? Okay, I can't see it. So we lost this particular part. Then we have again another N-terminal here, do you agree? And again this reagent is attacking here. Again we cleave it, now you get another structure called, something looks like N. Then you analyze it again by HPLC. So like that you will start degrading them one by one, right? And at the end of the day you will realize, okay, this is the final sequence of my protein. Looks really a lot of work, right? Imagine if you have 500 amino acids, you have to do a lot of work. However, this can be done automated today. So it's not that complicated compared to, I think I don't remember, it was in 1960s or even before. Now it's kind of automated, we can actually do the sequencing analysis. But to the best of my knowledge or my experience, I haven't read any paper they have tried to do the primary sequencing of some of the amino acids. They must have done it before in the olden days, but I don't have any collaborators who is doing this particular work. This information might not be necessary for you also in some cases. Okay? Does that make sense? How do we determine the primary structure of a protein? All right, now let's move on to little bit. This is something we use a lot in food science. That's called determination of the secondary structure, because the secondary structure determines the stability of the protein. How stable is your protein, and how stable is your tertiary structure is also dependent on the secondary structure. So we often measure the secondary structure. Why? Because food is often processed. Do you agree? Food is often processed. What is mean by processing? We apply light, we apply heat, okay? We applied shear forces, we apply pressure on the food. All this processing is done on the proteins, right? And when you do processing, the protein will change its structure, right? And remember my very first class, the secondary structures are stabilized by something called hydrogen bonding. Do you remember? So the hydrogen bonding is the one that stabilizes the secondary structure. Hydrogen bonding most commonly. That's of course there are some other weak interaction, but hydrogen bonding is one of the interactions that stabilizes the secondary structure. Now we all know that the hydrogen bonding is temperature dependent, right? If you heat it, hydrogen bond breaks. Just to give an example, like let's take water, right? Water contains extensive hydrogen bonding. Do you agree? Water exists in three forms, yes? Ice, solid, liquid and gas, and the three phases of water varies depending on the extent of hydrogen bonding. In the gaseous you don't have much hydrogen bonding, so the molecules go apart, so it becomes gas. In liquid you have a hydrogen bonding, so that brings the molecules together, so it becomes liquid form. In solid they are tightly packed because of an extensive hydrogen bonding networking, then it becomes ice. So hydrogen bonding determines the phase of the structure basically. Remember that one. And hydrogen bonding is also heat sensitive, it can be broken. So since you are processing food, there may be the structural changes, there may be the secondary structure change in the protein, and that information can be brought by FTIR spectroscopy. Does it make sense? Why are we using? Because often food is processed, and not only that, sometimes proteins interact with other biomolecules that can also change the hydrogen bonding and the structure, that can be also evaluated by FTIR. I'll give some examples later. Okay. Now I'll show you how exactly the hydrogen bonding is present in protein. This figure is not so beautiful though. You can see this is a carbonyl group. Can you see here? C double bond O, the carbonyl group. And can you see an NH, right? This is from another amino acid, the NH, right? And there is a hydrogen bonding between two, right? Electronegative and electropositive group, there is an hydrogen bonding, right? And that pulls the molecules together to make a bending or coiling and so on, right? That's the hydrogen bonding. Now because of changes, this hydrogen bonding can be broken. When the hydrogen bond broken, now the bent structure can be, the coiling might change, right? Originally it was nicely coiled, now it's maybe weakly coiled, and that indicates the structural changes of the protein. To evaluate that one, we are going to use FTIR, right? Have you studied FTIR spectroscopy in analytical chemistry? How many of you study FTIR? Okay, there is only two. Now I'm a little bit worried to what level information I have to give you about FTIR. Okay, FTIR is a spectroscope. I don't go deep into the spectroscopic, the principles of IR, but I'll just mention, since you guys don't know, I just mention how exactly IR works. Okay, IR is a radiation. Do you agree? It's a form of energy. IR radiation, similar to light, the visible light. There's something called IR radiation. Visible light is having wavelength up to 800 nanometer, and IR will have a little bit higher than that one. IR is also called as a heat radiation, just remember that one, right? And molecules will have a property to absorb light, right? Molecules likes to absorb light, And some molecules absorb some types of light, some absorbs UV, some absorbs visible, some absorbs IR, some absorbs some other types of light. It is a property of the molecule depending on their electronic configuration, right. Now, let us take a water molecule as an example, okay, and this molecule can absorb IR radiation, okay, and because it absorbs IR radiation, it can have a vibrational transitions, it can vibrate, right. What do you mean by vibrate? The OH bond can be stretched like this one, just to give one example, and this there is a bond angle here, can you see, and that bond can bend like this, like a scissor, or it can wag like this one by absorbing the IR radiations, and that is called the IR transition. It is very specific to particular molecule, right. Depending on the stretching, bending, wagging, scissoring, different vibrational transition, you get different type of IR spectra. Is the most simplest way to explain IR spectroscopy in my world, right. I am not going into detail, it is not my today's topic. So, just remember that molecules absorbs IR radiation and they vibrate, and the vibration will give you IR spectra. Keep that in mind, those who do not know IR spectroscopy, all right. Now, we have C double bond O and we have NH, can you see here? So, the peptide bond, especially in the backbone, the peptide bond that is over here, can you see, right? You have NH and C double bond O, can you see, and C double bond O, the bond can be stretched, okay, and the CN bond can be stretched, and NH bond can be wag like this one, bending, right. So, there is a stretching and bending vibration, and that vibration will give you a specific IR spectra, okay. If there is a change in the structure of the protein, the vibrational spectra will also change, and the change in the vibrational spectra is directly proportional to the change in the secondary structure of the protein, because we are particularly focusing on the peptide bonds here, right. That will change because when you break hydrogen bonds and something, their local environment changes, then there is a change and the spectra is changed, and that gives us a direct information saying, okay, we are breaking the secondary structure of the protein. Is it too complicated? Did you get the point? Should I repeat? Should I repeat? Okay, you are telling yes. So, IR is a spectroscopic technique. Remember one thing, IR is a radiation which absorb, some of the molecule absorbs IR radiation. When they absorb IR radiation, they will have a vibrational transitions. That means the bonds can be stretched, the bonds can be bent, the band can be wagged, scissoring, that many things will happen, and bending and stretching will give you an IR spectra, right. The IR spectra is very unique for particular bonds. For example, if you have a OH bond, you have a bending, stretching vibration, you have a bending vibration, you get a particular vibration, right. If you have a C double bond O bond, there is a particular vibration because of stretching, and they absorb IR radiation in a particular region, right, and that will give us a different types of IR spectra, right. When the protein changes its structure, the bending and stretching transition will change because of the change in the local environment, and that will give you a different vibrational spectra, and that will help us to understand protein secondary structural change, okay. Especially in proteins, we are talking about amide bond. Please note that there is a lot of other bonds in proteins, but the characteristic bond for a protein to do the secondary structure is called amide bond. This is amide bond. We also call it as peptide bond, they are one and the same, okay. There you have two transitions, there is a one here, the other here and the other here, and this stretching is called amide 1 transition, amide 1, and these two, the bending and stretching, this one here is called amide 2, this one. I hope I have a spectra. Yes, can you see? These are the two spectra of protein, amide 1 region. Basically the amide 1 region is this one, can you see this part, okay. Amide 1 region is this part where the stretching is happening, and then there is an absorption happening at around this particular wavelength, can you see, sorry, wave number here, make sense. and if there is a change in the bending of NH and CN, then the amide to the spectral pattern will change, because this particular vibrational transition will absorb in this particular region. Please note that it is very unique for the protein. It looks like a simple two peaks. No, it is very unique for the protein, because the shape of this peak and the position of this peak determines the structure of the secondary and the tertiary, determine the structure of secondary, determine the secondary structure of the protein. Make sense? So, the shape and the position, they are very, very important. Okay. Just to give that example here, look into this one. I have IR spectra of two different proteins here. The red one is protein number 1, and the blue one is protein number 2. Maybe the red one, the protein is unheated, the blue one is the same protein heated. Now, if you merge them together after getting the IR spectra, we take the IR spectra using this equipment. Okay. This equipment is used to get the IR spectra. You get the spectra like this one. Now, just by superimposing them, you get a lot of information. It looks very simple, but it is very actually complicated. It says, okay, there is a difference. Can you see here? Okay. And also, can you see the shape difference here? There is also some other part here also changing, but they are not very related to proteins, but yes, there is a difference, right. So, there is a difference in the shape, there is also difference in the position, the location, right, and the shape and the location of these particular the peaks will give us a lot of information about the secondary structural change in a given protein, all right. So, now I just to give more, a little bit more information about secondary structure is made up of alpha helix, right, where you have amino acids are coiled because of the intramolecular hydrogen bonding between C double bond O and NH of different amino acids. They pull together, that will help us to coil the proteins like this one, right, and they give the characteristic absorbance over here 1652. Okay. Then if you have a beta pleated sheets in proteins, right, and they have a characteristics over here, can you see? Then there is something also called random calls. Basically, they are always random. Difficult to say what is their folding and something weird ones, and they show characteristic IR peak over here, right. And these three information will be taken in the IR spectra, right, in addition to the shape, and that will be deconvoluted, and that will give us the information about what is the percentage of alpha helix do you have. What is the percentage of beta sheets we have? What is the percentage of random calls we have? The change in the percentage of these things will talk about change in the secondary structure. Okay. Please note that percentage calculation is not done often manually, it is always done automatically. There are some softwares, they will quickly deconvolute these peaks and peak shapes, and that will tell you, okay, now you have 25% of beta sheet, now you have 60% of alpha helixes, you have only 0% of random coil. Okay. You can see that there are 3 proteins here, there is a difference between their beta sheets and alpha helix, and that will give us information about the secondary structure. Please note that this is not done manually, it is all automated today. You can perhaps do it, I never did it myself, but there are dedicated software to do this deconvolution. All right. Any question until now? Yeah.
Sir, if we have a FTR spectra, how we can visualize whether this is for beta sheet or whether this peak is for alpha helix?
That is basically, I mean, there is some chemometrics optimization for this one. The system is calibrated initially for these particular absorbents. They know that, okay, if you have 1652 and okay, that is corresponding to alpha helix and so on. So, it is all automated. I personally do not exactly know what is the algorithm they use it to make this calculation. However, it is purely based on the wave number and the shape of the peak. If you agree with me, I think you have a person behind you, she can also give explanation. She has done a lot of IR. You're welcome to.
Sir, as I have read number of papers regarding these FTR, everyone has written there is a vibration, there is a stretching.
Yeah.
But peak, when we see the peak, it is always same. Although there might be difference, someone is going up, some is down, but we are never able to finalize which one is for transition, which one is for stretching, which one is for vibration.
Yes.
And one more thing is that it is, I think, highly confused data.
Yes.
I never.
It's
satisfied
very
with this.
normal for a normal person. If you look at an IR spectra, I think it's very similar, and it looks exactly the same for me. But now, if I put, if I don't superimpose these three peaks here, these three IR spectra here, it all looks very alike, right? However, the chemometricians, however, the software can actually pick that minor difference and tell you there is a difference. And please note that there is a big table in the, if you look into analytical chemistry textbook, you have, for example, OH, you have a specific at, I think, 3600, and NH, there is a particular wavelength they absorb. Every single bond, you have a particular wavelength. There is something called IR fingerprint region and functional group region, and there is a lot of information about what kind of molecules you have. Because let's say one example, let's take a simple example of, let's say, maybe a glucose. Okay, glucose. Okay, and this is a simple example of glucose, and basically you have a OH group here, you have a CH group here, and there is a lot of other functional groups, right? And they have a different types of bending, wagging, scissoring. There's a lot of combinations they do. That will at the end of the day give you the unique IR spectra for that particular compound, and that is something is calibrated. It's already in the literature. We know how exactly the IR spectra of glucose looks like, because it's a combination of different functional group, and the functional group region is above 1500, am I right? About 1500 per centimeter you will see all the functional group, OH, NH, C double bond O and something, and below that one you will get the fingerprint region. It's extremely unique for a particular transition, and combining functional group and fingerprint region, we determine the structure of the protein. It looks very, I mean, if you keep two IR spectra together, it looks exactly same for a normal person, but for the IR specialist, they say a difference. And she has a point to explain.
Yes, I want to answer this question of him, that he asked that I'm not able to get which particular wavelength is denoting the vibration or the stretching, right? It has been decoded by many researchers that which particular wavelength is denoting alpha helical structure, random coil structure, beta turns, anti-parallel beta sheets or beta sheets, intramolecular and in fact intermolecular. Right. So based on that, we can calculate using Origin software that you told that the percentage can be calculated. Yes, we can do calculate that, and that percentage gives the minor differences of every particular protein we have extracted.
Yes.
So on the basis of the percentage also, the strength or the functionality can be decoded of a particular extracted protein.
Yes.
So that's the answer of this.
It's basically the instrument is calibrated for particular vibrational transitions. Yeah.
I have used this Origin software to deconvolute these peaks, but as a food technologist, I was never able to differentiate what is the impact of the different treatments. Although I have used in, I think, number of papers that this fingerprint region, there is no data available on fingerprint.
Yeah, fingerprint
region.
is very, it's not for the proteins.
And
it
more
about
the amide A, amide B, amide C, there is no information available on that section. Only people have used 700 to 1600 this. They have used software. There are some steps you have to follow, and you will get this calculation. But to know the base, there is, I think, a lack of knowledge. I have never been able to find.
Yes, I think one thing I must tell you that IR alone, this is the first point I mentioned to you earlier, one technique will not give you complete information about the protein structure. No, these techniques are actually complementary to each other. So by just measuring IR, I would not say, okay, this is the secondary structure of the protein. It's impossible. Okay, then I go for CD to complement my data. Okay, CD is also showing the similar trend. Then I say, oh, maybe I'm doing right in IR also. And to answer your question about the complexity, IR is also spectra looks simple, but there is a lot of information in each peak. That's what I want to highlight. And to answer your question, I fully agree with that. Researchers often confuse you in research article just mentioning the different percentages and different wave numbers. They just say what they have seen on the spectra. You don't get much out of it. However, it's a fantastic tool to still measure the secondary structure of the protein. It's more about how do you complement, how do you deconvolute IR spectra to get more information. And there is a specialized branch called chemometrics in food science, and they do this job in a really nice way. And normal people like me, for example, I am a chemist, I know IR spectra to use for organic molecules, not necessarily for proteins. For me, it looks a little bit fishy, but for them, no. They can actually do a deconvolution a lot. It's another branch basically. I don't cover all these things, but I fully agree with you that the only superficial information is often provided in research article about IR, but it's still a fantastic technique. It's a very robust, never gives a lot of errors, but it's still there's a lot of disadvantages, but I can take it later. But I think we can still use it as a complementary technique to find out the secondary structure. It's my personal opinion. But the way you describe the data is very important, because the deconvolution what you do here, it should not be very personal, right? If she deconvolutes and gives, I have 65% in origin software, and you use another software, it shows, oh, maybe 63%. Oh, no, my structure is stable than yours. Maybe we conflict each other, and there it becomes more about the chemometrics tool doing things in a right way. This is all important, right? But I think in our case we have a specialized software, and they do it automatically by click. It tells you how this percentage. I never went deeper into that, how exactly the software calculates this one. I also myself use the data which is given directly by the computer, but I have some good colleagues. Basically, they do it in a very different way. Just to let you know. All right, let's move a little bit further. One of the interesting thing is that IR not necessarily can be used directly to determine the direct structure of the protein, the secondary structure. Rather, in food science, it can be used to find the difference, right? Untreated and treated, polyphenol added, polyphenol not added, heated, not heated, ultrasound treated, not ultrasound, not treated. Those differences can be seen very nicely by IR, right? Maybe it's very difficult to say the real secondary structure using IR unless you are a good person with all the software skills, but a normal person can actually see the difference between the two treatments using IR. What happens if I pasteurize my protein, right? What happens if I do UHT treatment of my protein? Those small differences can be easily seen in IR spectroscopy, because the heat is directly proportional to change in the protein structure. Change in the protein structure is directly proportional to the change in the secondary structure, which is directly proportional to the change in the IR amide 1 and amide 2 band. That is something I have shown you here. IR is very sensitive to temperature. Does that make sense? So I recommend you to use IR if you really want to see the difference between two samples in food science. Okay. One of the biggest disadvantages of IR for protein analysis is, especially for food samples, is water. Okay, that I have to highlight this particular point. For normal other people's samples, it's not a big problem, because let's say organic people, they extract things in hexane or toluene or something like that, and they can dry it, and they can completely remove water, and they can dissolve it in another organic solvent which is not IR active, and then they can measure an IR spectra. For food scientists, is always a problem. Why? Because almost all of our samples contains water. Do you agree? There is no food samples contain, even if you freeze dry, as soon as you remove from the bottle and you open, it absorbs moisture from the air, it becomes wet. So it's very difficult to find 0% water containing food samples, almost impossible. Proteins are always high, most of the proteins are hygroscopic, they absorb water molecule here and there as soon as you open the cap. And that gives you biggest challenge. Why? Because the water has a particular absorption in this region, and this region is also the region for the amide 1 and amide 2. That's where the protein absorbs. Now that can superimpose your data and that might mess up your data analysis. That is one of the biggest challenges. Maybe you should answer this question, how do you solve this problem often? Can you give her a mic? Yeah, yeah.
If the water is present in the sample, it will hydrate
Yes.
the molecules, and the orientation of the functional groups will be changed. So we'll not get the native structure we want
Yes.
for our analysis, and
Yeah.
there will be interference with the presence due to the presence of that
Yeah.
water molecule.
Yes.
Right. So to omit this, we have to dehydrate the sample completely.
Yes.
Either we have to lyophilize the sample.
Yes.
In the absence of, means the temperature should not rise, because we
Yeah.
are doing the protein analysis at high temperature. It will denature and the secondary structure will change.
Yes.
Or either we go for desiccation for at least 1 month.
1 month.
Yeah.
Now look at that's the point I wanted to mention here, is that how difficult it is to remove a water molecule from a protein, because they are actually hydrated in the sense that the water molecule can be trapped within the structure of the protein, and the trapped water molecule removal is a nightmare basically, and that can interfere your analysis. But I also heard from my colleagues that they can still deconvolute the spectra, removing the water contribution from the protein contribution, because there are a lot of tools for that one, what we call deconvolution techniques, and they can still get the information, but that requires next level of knowledge. Do you have a point? Yeah.
What I done means I dry it in vacuum freezer,
Yeah.
vacuum thing, and after that taking sample I will rush to that IR sample
Run and
the
put it in the
hydrophilic nature, due to the hydrophilic nature, I will
Yeah.
just do, okay, it's a fish collagen. It will be, it is not that much hydrophilic nature, but it's little bit hydrophilic nature. I will just run the sample in that.
So what she is telling, that you do the freeze drying or lyophilization or vacuum drying and just run to the IR machine quickly, open the cap, put it, close it, and just press the button on. Probably you solve the problem of interference. I agree with you. That's what most PhD students does. They are all their life is in a hurry always, right? So I agree with you, but this is one of the challenges of IR, just to let you know. Okay, there is a lot of advantages. I don't know whether I have a slide. There are a lot of advantages here. What is the advantage according to you using IR? Huh? It's chemical free. Basically, that's one of the important thing, is that IR is one of the easiest technique, right? You have your sample. There is no sample treatment except drying, right? You can also use liquid sample if there is no water basically. I mean, you can use another liquid, D2O and other things, and you put into IR and press the button. Within a couple of minutes, I mean even a seconds, you will get the IR spectra. The scan speed is so fast, within a couple of minutes you will get the IR spectra. No sample preparation, no addition of solvent, no filtration, nothing is needed. Put it, you get the data. That's the beauty. You require in the milligrams, exactly. Those you don't need something a very high concentrated. But for the food scientists, especially for the protein scientists, samples is not a problem. We always have a lot of proteins in our hand, right? So IR getting IR is very easy for us. Actually, that's true. But that's also one of the limitations I want to mention. IR cannot work if the concentration is too low. You get a very weak signal, because IR intensity is also concentration dependent. If you say a concentration, if you have a, let's say 0.001% protein, you get a peak like this one. 0.1% you get the same thing like this, and then the peak intensity changes depending on the concentration, and some people use this also for the quantification of the protein. I don't believe it strongly, because it has its own problem. People claim it can be used for the quantification, because IR intensity is directly proportional to the concentration of the protein. But there is a lot of problems. But just to let you know that if you have too little concentration, you don't get a good IR spectra. That's another problem. Maybe the water will overshadow the peak of your protein or your analyte of interest. Yeah. Any other? Yeah, she has a point there. Yeah. 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 You can reuse. But do we still use the KBR pellet ones? Okay, good. Yeah. They use, yeah. I have used it when I was doing my masters, but now we don't use it any longer, at least plate, yeah. Absolutely, yeah. Exactly. That is not possible. But basically, in general, it's a non-destructive technique. If you use this ATR type of machines, that you can reuse the samples. But if you add KBR pellet and if you crush it, and then you lost your sample. She has a point, yeah.
Sir, actually we have discussed that amide 2 is based on NH stretching vibrations.
Yeah, NH and CH, yeah, yeah.
NH and CH vibrations.
NH bending and NC stretching.
So is that together or
they say together.
they say together?
Yeah. So basically, I think there is something called amide 3 bond also, which is
Yes.
actually combination of these two.
These two.
Yeah, yeah, yeah.
So amide 2 is generally based for NH, right?
Yes.
So and in the same case, amide A and amide B is also NH. So how do this be differentiated?
I don't know this question. I don't know the answer for this one. Can you repeat this question?
Amide A bond and amide B bond is characterized by NH stretching vibrations.
Yeah, exactly.
The same way amide 2 is also done. So how do we differentiate it? Like
I
how
don't know an answer to this particular question. I'm sorry. Do you know? I never thought about it. Oh, they are very different. No, she's adding.
No. It's not about amide 1 and amide 2. It's about amide 2, amide A and amide B.
Yeah, exactly. Amide 1 and amide 2 is very simple. It's basically they have a very distinct IR spec, this one.
Yes.
But do you have an answer? She has an answer? Yeah. In the table. Yeah, yeah. Okay. What she's telling that there is a table with all the peak numbers and you just have to find it and assign it. But I'm sorry, I don't remember that one, to be frank. That's a good point. We will look into that later in the break. Okay? Okay. I don't recall the number. Okay. 13, yeah. 2200. Okay, we can look into the exact number for those things at the break. Is it okay? Good. All right. So I just give you some examples of advantages. Another advantage is IR equipment is always available in most of the laboratories. Not necessarily in the food science. You collaborate with your chemistry department, you get an IR instrument. So, and there is no much capital investment, and you don't pay for the samples also there because you don't use anything there, just except that sample chamber. But everything is free. Not free, but means you don't have to buy solvent and other things. But it's very easy. And that's another biggest advantage. Okay, now I give you some examples with the interest of time. I have to go a little bit faster here. This is an example showing that how different soy protein isolate, and when it's combined with the sugar units, glucosamines, and when it is reacted with an enzyme called transglutaminase. I'll explain transglutaminase in one of my lectures in the future. It's an enzyme, it will cross-link lysine and glutamine residues to make protein-protein cross-links, and they're trying to investigate exactly how that is changing the secondary structure. Just to give an example, how you can use IR to see the structural differences. As I said, food is never alone in food system. It's always, protein is never alone in the food system. It is always interacting with something, and those interactions can be visualized using IR spectra. Please note that when I teach you, I don't go deep into the individual spectra because you will forget it. I don't want you to learn all these things. I really want you to understand the take-home messages. That's it. Make sense? All right, now there is one more example here. What they were trying to do here is that they had this pea protein isolate and they used this glutaminase enzyme. You know that an enzyme called glutaminase. So you have a protein containing glutamine residues, which is having a structure like this one, glutamine, right? And which is having a positive charge. Now, if you use an glutaminase enzyme, glutaminase enzyme, and then it becomes glutamic acid. The glutamic acid is having COO minus. So basically, you can change the surface charge of the protein, right? And this might have a different solubility and different pI, and this might have a different solubility and different pI, and you basically change the functionality of the protein, in other words. And they were trying to understand, okay, when you change glutamine into glutamic acid, what is the secondary structure change? So, they could evaluate that in this particular region. You can see that there is some change. I do not go deep into what exactly they have seen here, but so just to let you know that if you modify the protein using different chemicals or enzymes, the secondary structure will change, and the change in the secondary structure can be evaluated with IR. All right, so now I will go little bit into tertiary structure. After this, I will try to give you a break. Is it okay? You are tired now? Yes, of course. We started at 9:30, so 1 hour. So after this, I will give you try to give you a break. Is it okay? All right. Now we move on to the tertiary structure determination. Remember, the tertiary structure is nothing but it's a 3-dimensional folding, and you have a disulfide bond. We have a hydrogen bonding. We have a hydrophobic interaction. Everything together makes protein to bend, and it will give you the tertiary structure. All right. So now there is a lot of different techniques to understand the tertiary structure. One of the easiest and the most common technique used today is fluorescence spectroscopy. Okay, it's cheap, rapid, easy. It has its own problems. I also tell you that one later, but it's an easy one. How many have studied fluorescence spectroscopy? No. Then again, I have to teach you the basics now. Okay. I will go again very briefly, please understand, because I expect that you know little bit basics before I teach these things. But since the audience does not do not know much about the spectroscopy here, I will cover the basics, but that will also take my time. I have to speed up my other slides. Just remember that. Is it okay? Good. There are this is a proteins, right, containing amino acids, right? And in IR spectroscopy, we mentioned about some of the bonds absorbs IR radiation and they start vibrating, right? And there are some molecules, they absorb UV light or visible light, and they can excite their electrons to higher energy state, and then they will give you fluorescence. Okay? There are 3 amino acids on protein, they absorb UV light very nicely. What are those? Tryptophan, tyrosine and phenylalanine. They absorb UV light. Why? Because they have aromatic ring. Can you see? An indole ring, a phenyl ring, a benzene ring here, right? Because of these aromatic rings, they absorb ultraviolet light. You know what is UV light, right? What is the wavelength? 200 to 400, that is a nanometer. And usually the molecules absorb a particular wavelength or the range of wavelength. For example, the tryptophan has an UV spectra something like this, okay, and the maximum absorbance is around 280 and to 295 nanometer. Okay. So they absorb a particular wavelength in the UV region, right, and when they absorb light they go to higher energy state. I will come back to that point, remember that point. And tyrosine can also absorb light, the UV region, and also phenylalanine, but when compared to these 3 amino acids, tryptophan absorbs light in a very high efficiency, that is the right word. The quantum yield for absorption of UV light by tryptophan is very high. So we use tryptophan residue on a protein, this is tryptophan for example, to understand the protein's tertiary structure change. Is it clear? Just to simplify things further, proteins contains lot of amino acids. Among that there is one amino acid called tryptophan, and tryptophan absorbs light in the UV region. Once it absorbs the light in the UV region, it can be excited and gives fluorescence. I will get back to how it gives fluorescence later, and that property is used to understand the tertiary structure. How? Now, okay, I already have this one. Can you see the this one? Different absorption spectra for this one. This is a tryptophan, the red one, okay, and it absorbs around 295 nanometer in this case. All right. Now I will show you a little bit about. I will show you something about how we can use this for the structure determination. Now this is a 3-dimensionally folded protein. Just you imagine. This is not chapati or dosa or something. This is 3-dimensionally folded proteins, okay, right? And tryptophan can be present inside the protein. That means it's called buried inside. Let's take an example here. This is tryptophan amino acid, okay, this is protein. It can be present inside the protein like this one, what we call it as hydrophobic region, because when you put this entire protein into water, the water is here, right, it's a hydrophilic region or aqueous region. The tryptophan can be present inside the protein. Now it's not accessible to water, can you see, because water is outside, tryptophan is inside, so now it is in the hydrophobic region. Now I am heating this protein, I am boiling the water, what happens? Bonds gets broken, hydrogen gas bonds get broken, protein will start opening up like this one. Now can you see whom can you see now? Tryptophan. What do we have around here? Water molecule. Now tryptophan starts interacting with the water molecule. Now tryptophan fluorescence properties changes depending on whether it is in this form or whether it is in this form, right, because the interaction with the surface, the microenvironment changes the fluorescence properties of tryptophan, and that is used to understand the tertiary structure of given protein, basically change in the tertiary structure. Please note, it is very difficult to say what is the tertiary structure, it is basically we determine the change in the tertiary structure. Is it clear? So tryptophan, if it is buried, it is in hydrophobic region, it has a different fluorescence property. If it is opened up, it has a different fluorescence property, and that is measured in the fluorescence spectroscopy. Keep that in mind. Now I am going to teach you what do you mean by fluorescence, because you guys have no idea about that, okay. So you know that nucleus in an atom, in a molecule, right, and we have somebody revolving around. Who is that? Crazy guy, electron, right. So these electrons are just circling around the nucleus in 3-dimensional orbital. That is all orbital theories you studied in your high school, forget about it for now. But I can mention these electronic levels like this, okay. Now I have a tryptophan, right. Imagine tryptophan in its outermost orbital, it has 2 electrons. Electrons are shown as half arrows, like, can you see? Now I take a UV light, I can take a UV torch, and it has a wavelength around 280 nanometer, imagine, okay. I shine this light on the UV, on the tryptophan molecule in solution or in solid state, whatever, in solution, right, and then the light falls on the tryptophan now, right. When there is a sufficient amount of energy, the energy is in the form of light here, the UV light, electrons absorbs the light, right. Once they absorb the light, what they do? They excite, they go to higher energy state because they have extra energy, they do not want to stay in the home, they just want to get out, that is what we do, right. I am more energetic, I want to go to my friend's home and do a party, right. So I take that will excite to higher energy state, right. Now the electron came all the way here, right. Then basically when you go to a friend's home, you enjoy there, but you do not sleep there, right, do you? Very rare, huh? You come back home, right, and that is called coming back to the ground state. But often you do not come back home directly, you go to some supermarket and have friends, another chat. So basically they come back to the another energy level like this by losing the energy in the form of heat. There is a lot of theory here, because I do not have time, I will only show you the basics, please understand that. So they lose the energy in the form of heat, okay, and then they come back to the ground state. While they come back to the ground state, they release the energy back in the form of light again, okay, and that is called fluorescence. I am super bad with my spelling, do not believe in my spelling, but understand what I say, okay. So fluorescence, is it clear, right, and that you can get as a spectra. You have an absorption spectra and you have an emission spectra, right. This is where the electrons are absorbing, you get a spectra. These are electrons are emitting back the energy, that you get a spectra. So it is called absorption spectra and you get a emission spectra. The emission spectra is basically a fluorescence emission spectra, and that depends upon where exactly tryptophan is present. I will show that in later case. Is that clear? Now I repeat this one again, is an important concept. So electrons are usually in the ground state. By absorbing the light energy, UV light for tryptophan, it goes to higher energy state. Once they go to higher energy state, they not necessarily come back to the ground state directly, they lose the energy in the form of heat. That is called vibrational relaxation, or in chemistry, but I do not teach you that one. A small amount of energy is lost as in the form of heat, and then they jump back to the ground state by losing the energy back, and that is called fluorescence. Okay? It is actually a unique property of a molecule. Every molecule absorbs a particular wavelength and they emit at a particular wavelength if they are fluorescence active. That is a very unique. Tryptophan absorbs at 280, emits at 340, for example. If there is a change in the microenvironment of tryptophan, excuse me, if there is a change in the microenvironment of the tryptophan, then the absorption intensity, sorry, emission intensity and the wavelength will change, and that we will use for the fluorescence spectroscopy. Is it clear? Any question in this part? Please raise your hand. Did you understand what is fluorescence and fluorescence spectroscopy? Okay. There is also something called phosphorescence, but that I will teach you some in my next life if I come here. Okay. All right. So now just to give my explanation here, this is an absorption spectra of tryptophan. Can you see? Okay. And this is an emission spectra of same tryptophan. This is absorption. This is emission spectra. Now, absorption spectra, the maxima is around, let us say, 295 nanometer. The emission spectra maxima around 345 nanometer. There is actually a difference. Can you see? Can somebody explain why there is a difference between absorption spectra and emission spectra? Why there is a difference in the wavelength? I make my statement again now. The emission wavelength is always lower than the absorption wavelength. The emission wavelength maxima is always higher than absorption wavelength maxima. Why? I give you a hint here. Look into my Jablonski's diagram. Yeah. Exactly. Can you give her a mic?
While coming back from the excited state, it is losing as heat and then light. So we are only detecting the light as the emission spectrum.
Exactly. Some portion of the energy is lost in the form of heat. Energy is lost in the form of heat. You know that energy is inversely proportional to wavelength, right? Who told that one? Einstein or Max Planck? I have no idea, right? Energy is inversely proportional to wavelength, right? So basically you lose the energy, so the wavelength is increasing, right? Because you lost some part of energy here, you absorbed this much, right? And you lost this much as a heat, and the remaining is emitted, right? And that is the reason why the fluorescent spectra always having the higher wavelength region than the absorption spectra. Does that make sense? Especially the maxima. All right. So now when the tryptophan is in the buried region, and when the tryptophan is in the exposed region, they have a different absorption spectra. Can you see here? The absorption spectra of tryptophan varies depending on their microenvironment, how they are sitting on a protein, right? I have some small explanation here. Now, if the tryptophan is buried in the hydrophobic region, okay, basically they will show blue shift. What do you mean by blue shift? The emission maxima is towards the left hand side. Imagine the original emission maxima is, let us say, 345 nanometer, okay. If the tryptophan is more and more buried now, then the emission maxima will go in the left hand side, right. Now the same tryptophan is now under open condition like this one, exposed condition, then it will have red shift. That means the emission maxima will go in this direction. And the change in the emission maxima or the intensity and also the position of the spectra will talk about the folding of the protein, or in other words, three-dimensional structure of the protein, in other words, denaturation of proteins. Have you heard about denaturation? Proteins will be denatured when you have when you do the heat treatment. Protein will be denatured when you do STS treatment. Protein will be denatured when you treat under urea. All this thing can be investigated using this technique, right, because of the tryptophan's change in the emission properties. Okay, I told the right thing, right? Aqueous, you will have a red shift, When it is buried hydrophobic region, you will have a blue shift. Is it clear? Any question until now? Yeah. What's that? Again? Yeah, it's basically, just to summarize it again. So now basically imagine the original, this is your protein, untreated protein for example, and you take a fluorescence spectra of this untreated protein. You ignite it at 295 nanometer and you measure the emission spectra. The emission spectra will look like this one. The emission spectra will look like this one, right? This is the emission spectra. Imagine the maxima is like 345 nanometer. Okay. Now you did the heat treatment of this particular protein, right? And then it will open up, right? Now the tryptophan is exposed. Now it's interacting with the water molecule, right? Change in the microenvironment of the tryptophan will change the emission intensities and also emission, the type it emits, right? Then basically if it's opened up, the emission maxima will come like this. So that is called red shift. I think it's in chemistry you studied about bathochromic shift, hypsochromic shift, otherwise called red and blue shift, right? So this is called red shift where you get a higher wavelength emission. So it may be like 365 nanometer now, just to give an example. Does that make sense? Now for a reason you did some experiment, now the tryptophan is more buried, right? Compared to the previous one, it's completely buried now, then you will get the maxima over here. It comes to 302 nanometer, imagine. And that information will tell us about the protein 3-dimensional structure, denaturation, also renaturation. All right. Just to give some more examples here, any other question? Just to give some more example how food scientists are using tryptophan fluorescence to determine the protein tertiary structural change. I use the word change, please remember, okay?
The mechanism of means the chemistry behind your telling about the blue and red shift, can we link it to the fact that when it is being shifting from hydrophobic to hydrophilic, means more of the bonding will be there, so more of the energy is required, so the higher the wavelength is there for the absorption, and when it is shifting from hydrophilic to hydrophobic, so cutoff of the bonds is there, so that's why it is going to the lower side. And
it's very difficult to say that way.
Is there any connection?
No, I mean it's yes, and that information is not readily available. It's a very complicated phenomenon because there is a lot of transitions are involved in here. The vibrational relaxation, the loss in the form of heat, it's all related to the micro environment of the protein. Basically how the additional molecule are actually interacting with the protein, and that has an influence on excitation and emission behavior of the given molecule. And to the best of my knowledge, I try to search the same thing, what is actually the real mechanism why we see. It's very difficult to find the actual reason why there is a change in the transition depending on the change in the microenvironment and so on. I don't have a clear-cut answer for you that particular today. Probably I need to dig a little bit more into the literature. However, it's something related to the microenvironment, basically how a molecule is interacting with the other molecule, and that changes the surface properties, that changes the polarity, and that changes the emission behavior. Yeah. Yeah, I'm not very sure how I can explain that one, because basically in fluorescence spectroscopy we do not make and break any bonds. It's more about the excitation of the electron, and the emission is purely dependent upon like if you look into this Jablonski's diagram, like there's a lot of vibrational state, right, and a lot of intermediate energy states, and basically now you excite into this particular region. There's a lot of possibilities that for to come back to the ground state. It can come back from here to here, it can come back from here to here, it can come back from here to here, and that will give you different spectra basically. And this changes is depending on the microenvironment, to be frank. And there is no making and breaking bond in principle here because that is not that energetic in fluorescence part at least. It's mostly about the energy transfer in my opinion. Yeah. All right, so now these guys were trying to do the P protein isolate, and what they were trying to do is they were trying to investigate the ultrasound treatment. Remember the ultrasound assisted extraction? I mentioned it's a gentle technique, but there is a heat is involved, right? And some people also say that ultrasound generates hydroxyl radical. They are oxidizing in nature. They can oxidize the proteins. When the proteins are oxidized, the structure is changed, and that can be evaluated here. And these guys are trying to do that one. You can clearly see that imagine this is increasing the ultrasound treatment intensity. It's basically increasing amplitude and time here. They can clearly show you that, okay, you can see two things here in this spectra. What are those? What are the two differences you can see here in terms of fluorescence spectra here?
Yeah.
Yeah, the change in the intensity of the peak, and the second one is shift in the lambda max. Please note that these are two things always you look at the fluorescence spectroscopy. Usually the area does tell something about that one. If you know the deconvolution technique, is basically the intensity of the peak, which is again directly proportional to the area, and the shift in the lambda max, that talks about the protein conformational changes. As I mentioned, if the protein is more and more exposed to hydrophilic region, aqueous change, you will have the red shift. If the protein is more and more exposed to hydrophobic region, you will see the blue shift, right? And this paper, and some people also claim that if the tryptophan is in the hydrophobic region, you will have increased intensity. If tryptophan is more in the hydrophilic region, you will have a decreased intensity of emission intensity. Okay, but I tell you that don't believe much in the intensity, but always try to look into the lambda max, and that gives you the more clear information, especially when you have impure protein samples. In food science, it's a problem, right? When you have an ultra pure protein, you can talk about both intensity and emission maxima, but if you have an impure proper, it's very important to look into the change in the lambda max, because the change in the intensity is also depending on many other factors in fluorescence spectroscopy, especially the interferences. Okay? And this is another example where they are trying to investigate the protein polyphenol interaction. You remember my last slide, somebody was talking about the color and so on, right? Plant proteins having the biggest problem is the color. They are always colored, right? And most of us believe that this color is because of polyphenols or anthocyanins or tannins, right? And these guys were trying to investigate. What they did was they incubated pea protein isolate with quercetin. Quercetin is a phenolic compound, right? And they saw, okay, increasing concentration of quercetin, they saw decreased fluorescence intensity, and they also saw change in the lambda max. Can you see? And that indicates them that, okay, now how quercetin is interacting with protein, and also whether the protein three-dimensional structure is changing because of this interaction, and that knowledge can be brought by this one. Okay. Before going to the next slide, and can you tell me little bit about the advantages and disadvantages? I taught you a lot about fluorescence spectroscopy. What's that?
Specific to some amino acids.
All
the amino acid will not emit the light.
All the amino acids will not absorb the light in that particular region, and all the amino acids are not necessarily fluorescent. It's only 3 amino acids are fluorescent. That's a one good question, but actually it's a good thing. It's not a bad thing.
Identify it easily.
Exactly, and because then you don't have a problem with too many spectra. If everybody absorbs, you get a very complex spectra like an IR ones, right? Right, we don't want that one. It's nice that only tryptophan and sometimes tyrosine and tryptophan phenylalanine is absorbing. So it's actually very specific and selective. That's a good thing to have. If every single amino acid absorbs, you have a very complicated spectra. Maybe it's a good thing for somebody, but for me it's very complicated. Yeah, good point. Anything else?
Sir, how will you usually analyze the fluorescent image, sir?
How do we analyze? We have a fluorescence instrument.
Okay.
So
after the data, how
after the data is basically, again there is a deconvolution techniques, and again there is a software to do this one.
What are the softwares?
Oh yeah, it's very difficult to say. There is hundreds of different softwares for that, and you can build your own software also. Basically the instrument will give you by default, every fluorescent fluorimeter will give you 2 spectra. which is called absorption and which will got emission. That's by default you will get. Usually you get, you can do the kinetics, you can do the heat treatment inside the fluorescence equipment, you can change the temperature, you can continuously acquire the spectra, right. The instrument will give you the spectra directly like this one, and also as an Excel sheet .csv file. But the modern equipment can actually do the landscape measurement. That means you can start exciting at different wavelengths. I talked about only 295 nanometer. You can start exciting tryptophan from 280, 281, 282, 283, 284, and you will get a landscape of the fluorescence, like a 3-dimensional spectra you get. And that can also be used to get more information. There's a lot of way you can use fluorescence. I don't go detail into that one. Yeah.
Sir, if a protein is deficient of tryptophan, then this technique might be useless?
Yes, if the protein is deficient of tryptophan, you can never use this one, because you can use with tyrosine, but tyrosine quantum yield is very, very weak. Phenylalanine is always nil, so it's very, you will get a weak signal. However, there is an extremely rare case where you will find a protein devoid of tryptophan. It's an extremely rare case. Almost all plant proteins have a tryptophan, maybe to a lower extent, but there is some. It's an extremely rare case. Yeah.
Yes.
Okay, can you look into one of the disadvantage looking into just this figure? The fluorescence has have a biggest disadvantage. Just look into this figure. Interaction with the other molecule. Usually, if someone else is absorbing at the same wavelength, then you will have a problem. You have a competitor in the solution, right? It's always the case in food science, right? Food is never a single molecule, I'm telling. You have protein with tryptophan, and you also have a polyphenols, imagine, right? Tryptophan is absorbing a light. Absorption is not a big problem because you can always put a lot of intensity, no problem. But once its emission happens, the emitted light can be absorbed by a polyphenol, which we call inner filter effect, right? And then everything will change. What you say and what you see and what is actually happening is different, right? The fluorescence spectroscopy data is a big mess in the literature. I'm sorry to tell this one, okay? Fluorescence spectroscopy in food science, I'm not telling in other part of the world, food science is a big mess, because sometimes they ignore the presence of some other molecules in food system, and they don't say about what these molecules are doing in fluorescence spectrum. So I'm openly telling now that don't believe fluorescence spectra data if they do not clearly explain how exactly they were measured, how exactly they have deconvoluted the data, then don't believe in that data. And don't use fluorescence if you don't know exactly what you want to do. If you have a complex samples with polyphenols or absorbing molecules, you have to be extremely careful if you use fluorescence. It's not just because I have a fluorescence equipment, I can easily measure emission excitation, I get a beautiful graph, I can publish it. That's wrong, right? You need to understand what exactly you're doing, especially if you have polyphenols or vitamins and other absorbing molecule, because polyphenols, especially flavonoids, they absorb around 330 nanometer. Fluorescence emission is also in this range. Can you see? You have a problem. So emitted light can be reabsorbed even by the same protein also sometime. So that will give you a big problem. So what you say is not true. Okay. So we must take a break at the interest of time. So we will take a break now, and we will come back after around 13 minutes. We will come back 11:15. Is it okay? We take a break 11:15. So I'll leave back to the previous slide here. So I could not cover one of the topics before I take the break. So I will take this one and I'll put it in here, and we have a break now. Okay. Yes. Ready? Ah. All right, welcome back, and we shall resume. So we have been discussing about spectroscopic techniques that can be used to characterize proteins, and we have covered primary structure elucidation using Edmonds degradation or Edmonds degradation, and the secondary structure we discussed about FTIR, and towards tertiary structure we have been more talking about how we can use fluorescence spectroscopy to evaluate tertiary structural change. I've been using the word change because basically in food science these techniques are actually useful to evaluate the change rather than really finding the structure, simply because it's a bit complicated, because food is a complex matrix. It's very difficult to get individual proteins to actually determine the real secondary or real tertiary structures, because it's a challenge in food science. We are not producing ultra pure individual proteins. I mentioned to you about the complexity of the plant proteins, for example. It's a mixture always. Now we shall move on a little bit more sophisticated techniques, but not in detail, just to showcase what instrument we can use or what technique we can use to get the real tertiary structure of a protein. One of the most well established method is X-ray crystallography, what we call XRD technique. You can also use NMR to find out the real tertiary structures, but the most common technique today is X-ray. This technique is useful if and only if you can crystallize proteins into individual crystals, which is one of the biggest challenges, because not all proteins are crystallizable. If you can crystallize, organic molecules can be crystallized. If you go to organic synthesis laboratory, they try to crystallize their synthesized drugs or whatever, because they can actually talk about how exactly the structure look like. It's a very complementary technique to NMR at the end of the day. But crystallizing proteins is a nightmare. It takes a lot of efforts. If you somehow can crystallize individual proteins, then you will get a fantastic 3D structure of a protein, and that is something people use to develop the databases. You must be knowing that the protein data bank, PDBs and so on, where you get a lot of information about amino acid sequence and the structures of a known and unknown proteins and so on, they all often come from this kind of methods, that they do the crystallization, they get the structural information, they get the sequence information, then they understand the folding and so on. If you recall, there was a Nobel Prize last year on protein structural elucidation, if you remember. You can also use de novo sequencing and modern sophisticated artificial intelligence technique to find out the three-dimensional and also quaternary structure of proteins. XRD is a simple, not simple technique, it's a complicated technique. You get a protein and you just crystallize them, and then you get the X-ray diffraction data using an XRD equipment, a very expensive unit. I don't know, we have it in every institution here in India, but it's also a specialized instrument, is only found in specialized laboratories. And based on that, you can get the electron density. Basically, you know that the protein is folded like this, right? Every atom or every molecule, every atom, in this case amino acids, they have a special electron density. It's a unique for that particular atom. It's unique for that particular amino acid, and that density information can be captured using sophisticated software, and that will help us to develop the protein three-dimensional structure. I will not go any detail into this technique because there is a professor coming in the coming days who will give you an exclusive lecture on X-ray crystallography, how we can use XRD to determine protein structure. So it's not my job now, but I'm just telling that we can do these things using XRD. Is it okay? I think you're happy now. I'm not teaching you too much, right? So this is how the X-ray crystal structure look like on a computer, right, after deconvoluting everything. So these ribbons and all these things you can see, beta sheets and random coils, and so all this information often comes from XRD, the diffraction factor pattern. This is one of the nicest structure showing the quaternary structure of soybean corn glycinin. So you can, I think it's a beta heterotrimer, homotrimer. So you can see the individual protein subunits. Can you see? That is showcased as a different color. It's just an interpretation. Please note that protein does not have a real color. It's just to showcase you in the computer, on a computer screen, we show it in this way. Okay? And if you go to the PDB or some kind of a software, you can actually turn and tilt and see how exactly the protein looked like on a 3-dimensional level. I can't show it on my computer, unfortunately, on a PowerPoint, but you can actually tilt and see how exactly the protein looked like on a 4-dimensional way, right? And where is the space inside, where is the random coil floating inside. You can also pinpoint where exactly that tryptophan, the individual tryptophan is located. Oh, now it's buried or it's on the surface using this X-ray crystallographic technique. It's another structure of moringa. I just wanted to show that the protein can be as big as this in corn glycinin, very complicated. It could be also very simple protein. But to get this structure, crystallization is very, very important. It's a bottleneck often. So probably the other professor will teach you how to crystallize proteins. Let's see. Okay. Any question about XRD? I don't want to take a lot of questions here because, as I said, somebody will teach you. Is it okay? But I'm going to teach you one of the most interesting and important technique, that is STS page. A very simple technique, but gives you one of the most, it gives you the overwhelming amount of information about proteins. Doesn't matter whether it's milk protein, plant protein, animal protein, bacterial protein, fungal protein, you can use STS page irrespective of protein type. Okay. So in the previous slides, we are talking mostly about getting the structural information, but STS will give you more about the compositional information. It doesn't talk a lot about structure. It does in a certain cases, but usually it talks a lot about protein composition, means what type of proteins you have. For example, in my very first class, I mentioned to you about soy contains globulins, right? Corn glycinin and glycinin. Rapeseed contains nepin and cruciferin, and our pea protein contains some other type of proteins, including prolamines and so on. And how do we visualize them? How do we separate that different subclasses of proteins? For that, you can use STS page. I really want you guys to learn STS page if you get a chance. If you want to take a career in food science, especially in protein science, by default, you should know how to run an STS page equipment. Okay, this is something we expect if you want to be a PhD student in protein science laboratory. It's a very common technique, not difficult. If you get a hands-on skill, it's easy. I'm going to teach you how exactly STS works. Is it okay? In the next couple of minutes. Well, what is STS? STS is sodium dodecyl sulfate, which is an anionic detergent. Okay, STS is an anion. Anion is negatively charged, right? So it's an anion detergent. You all know that the detergent, what does they do? They clean things, right? Yeah. So basically, this detergent will clean proteins. In other words, it will denature proteins. Okay, it disturbs the hydrogen bonding or hydrophobic interactions on the protein. The protein is 3-dimensionally folded like this one, right? Now, if you add an STS to this one, the protein will become like this. Okay, they don't cleave the disulfide bond. Remember, STS cannot cleave the disulfide bond. It can only cleave the weaker interactions. All right, and that will denature the protein. That's the first step you have to keep in mind. Okay. What exactly we do here is that now you have a protein sample. This is our protein sample in a tube. Okay, and we add STS. Okay, and we also heat sometime around 90 degrees Celsius for 10 minutes, depending on the situation, and then the protein will be denatured like this, right? Not only the protein will be denatured, since it is an anionic detergent, it adsorbs on the surface of the proteins and it will give the net negative charge on the protein surface. Now, the protein will be negatively charged. Am I clear? STS will do two things. The first one, it will denature the protein, and the second one, it will give you the surface negative charge on the protein. Protein can have positive, negative, everything depending on the pI and the pH of the solution, but as soon as you add into STS, it will get mostly negative charge. Keep that in mind. All right. Now, I have a protein solution containing negatively charged proteins. All right. And now, I take that proteins, I just want to have some space, put it on a gel. This is a gel, okay, and the gel is actually made up of polyacrylamide, which is a polymer polysaccharide, polyacrylamide gel, okay, and the gel contains small wells like this one, okay. You can have a number of wells depending on the gel you prepare. You can prepare this gel in the lab your own, or you can buy from the from the supermarket, but from the chemical company. You can buy them, okay. So now I will load the sample to one of the wells here, okay. A small amount, we are talking about 6 to 10 microliters containing the protein amount of some micrograms only, it is not a high amount you are talking about. And the entire gel I am going to place in a beaker or a chamber containing some kind of buffer, okay. Entire thing will be placed in a chamber containing buffer. Now, this chamber can be connected to some sort of electricity, right. It will be connected to positive electrode and also it can be connected to negative electrode, something like this, okay, and that will go to an electrical system like this. Now, there is a switch here, right. I turn on the switch. What would happen? Okay, so as per the charge of the protein, it will separate. And which direction the protein should move now? Downwards, right? The protein will start moving in this direction. Why? Because remember, ST has given protein a negative charge and you have positive electrode in this one. We have studied in our physics that negative loves positive. So, it just wants to go towards the cathode, right, moves in this direction. So, protein will separate based on the charge, but is it only the charge? Molecular weight or the size of the protein also does matter. Who will run faster? Those who are having the less molecular weight or the size will move faster. The bulkier stays behind. Okay. Now, here it is basically a mixture of protein containing small and bigger proteins, and then you are loading it as a mix here, right. Because of the size and the charge difference, the bigger one will stay here and the smaller one will come here. So, basically based on the size you can separate protein as a band. Is it clear? Based on the size, protein will separate as a band. We call it as STS page protein bands, right. Does protein has a color? No, but when you do this one, can you see the bands with your naked eye? Exactly, because our eyes can only see apparently visible light, right, and the proteins does not absorb in the visible light. So, you can't see proteins unless you are God, right. So, what we have to do is that we need to make protein somehow colorful, and then after doing the electrophoresis, the whole technique is called electrophoresis, okay. We put that in a staining container containing some kind of a dye called Kumasi brilliant blue. I do not write the full name because I have no idea about spelling, okay. Kumasi brilliant blue, that is a Kumasi staining. Is it CU something, okay, I write some wrong spelling. I said you that I am a bad with spelling. There is a Kumasi dye. So, it is actually giving a blue, it is a blue dye actually, and it loves proteins and it attacks the protein, then you get the band as a blue color band. It looks exactly like this one. What we do in real life is that in parallel to your sample, we run a molecular marker. The molecular markers are nothing but they are the proteins with known molecular weight. The company will tell you this band is equivalent to 14,000 Dalton and this band is equivalent to 2,000 kilodalton, because the company has already measured it when you buy the band. So, we run the molecular markers in parallel. The molecular markers are basically a mixture of different proteins with different molecular sizes. So, that will also run along with your sample, and that will help us to understand which protein, what is the molecular weight of my protein. Now, you can see in this case my protein is this one, imagine. I can tell that, okay, My protein may be in this particular band, maybe between 31,000 to 45,000. I say maybe the molecular weight of my protein is 40-ish thousand. Do you agree? And that's how you use STS page to find out apparent molecular weight, not the real one, apparent one, approximate molecular weight you can actually determine. I use this word approximate. Don't tell anybody that STS can be used to find out the right molecular weight. No, it just tell you an approximate molecular weight. But that information is very important for us because we talk about big proteins and small proteins in food science. So that can be easily visualized by STS page. Very simple, right? STS page. Any question? Principle is clear. I repeat once again, we have proteins with different molecular sizes and we add STS on top of it. STS is an anionic detergent. It will first denature the protein and then it will keep the negative charge on the protein surface. Then we take this protein mixture and load into a gel called polyacrylamide gel, right? And put the gel into a chamber containing buffer and we connect it to the electrodes. Proteins is negatively charged and we have positively charged electrode on the bottom. So then the proteins start migrating slowly based on the size of the molecular weight of the protein, right? And then you get a separation and that is very useful for us. Usually we run it for 40 minutes, 50 minutes, 60 minutes depending on the time and the voltage basically. That's the only two parameters you can change. You can change the voltage and you can change the time of the run, then you can have a separation. There is no much to play in STS page unfortunately or fortunately. You always use a unique condition in most cases. Now I did it's my personal experience. One day what I did was I hooked positive to negative and negative to positive and then I turned on the switch. What happened? Protein, the protein will go upward and come to the beaker. Basically, this is the common mistake our master students do, including myself. I also did that one. We're in a hurry and we are just you have to hook the black and red wire. If you just to reverse it, instead of coming protein downwards, it goes above the well and you lose the sample. That's one of the common mistakes we do in the lab, right? Of course, you have to do those mistakes because then only you will learn, right? All right, so any question about the STS page and the principle and how we separate the bands? Very simple, right? Now, I'm just giving you some example how we are using in alternative protein world STS page. This is a nice STS page I found from this particular article. I have no idea what did I do here. Okay, good. You can see that how we can separate different soy protein subunits. For example, corn glycinin having subunits can be separated. The subunits are separated because when you have because soy protein has let's say corn glycinin, let's say it has two subunits, it has multiple subunits. Because of STS you add, the interaction between them is broken and the subunit becomes individual subunit and they migrate differently. Can you see here? So now STS can also be used to understand what kind of subunits a given protein has, right? And it also helps us to understand different kind of proteins. And you can also use STS for the more complicated stuff. This guy tried to separate all these allergens. Allergens are proteins. I'll talk about that later in other class. So you can also separate allergens and see what kind of allergens you have. But please note, one of the important thing is that by looking into the band, you don't get the information. Because the only information I have the marker here. It's all about finding back to the literature, right? That's very important in STS page. So you just see a bands and you just see, okay, molecular weight, what are those? Nobody knows that idea. You just have to go back and see the literature, who is telling, okay, this person is also getting the same band. Usually, a 99% chance that the STS page look alike, even if you do in this lab and the other lab, provided you use the same condition. The bands will not change their order. The order is always same. The marker will always move the same way. It's usually a very robust technique. So that will help us to understand, okay, this band belongs to corn glycinin, this band belongs to glycinin, this is a beta lactoglobulin, this is alpha lactalbumin. That information can be bought. So not directly, but indirectly. Is it okay? Clear? So this is a pea protein isolate. We can see that how they are separating corn albumins and corn vicilins and other proteins. And this is a nice STS page gel picture of a rapeseed protein isolate. It contains cruciferin subunits and nepin, a smaller protein. cruciferin is around, I think, approximately 300,000 Dalton, and nepin is around 14,000 Dalton. So basically that can be separated. Okay, and you can also use STS page to see the impact of protein modification. For example, you are doing ultrasound treatment, then because you may break the proteins, and that can be visualized, and you are using extraction. For example, we talked about salt extraction, we talked about alkali extraction, right, in yesterday's class. Now in the salt extraction and alkali, this is salt extraction and alkali extraction, maybe the protein extraction yield is the same or different. Not only the yield, the type of protein you extract is different usually. Maybe salt is selectively extracting salt soluble proteins, and alkali extraction does not care, it extracts everything. You want to see what is the difference between the composition. It's not always about the yield when it comes to food science, it's also about the composition. So the composition variation depending on the type of extraction, that you can use STS page. If you are doing some extraction studies, I really recommend, it's a nice add-on to your manuscript basically, just to see the quality of the protein you are extracting. Make sense? All right. So another important advantage of STS page is we can also determine whether there is a disulfide link in the protein, okay, particularly whether there is an intermolecular disulfide linkages. Okay. If there is an intermolecular, this is protein number 1, this is protein number 2. If there is an intermolecular disulfide linkage, that can be visualized by STS page. Can somebody think little bit about it? How can we do that? I give you 2 minutes to think. You can think alone, or you can discuss the with the person next sitting next to you. STS page can be used to visualize disulfide linkage. How? How we can use STS page to determine that? Here is an example of a disulfide bond. This is you can see the intramolecular disulfide bond here. I really want to show whether there is an intramolecular disulfide bond. How do I show that? Yeah. Is this room too hot for you? Is the AC is working? No, it is too hot. Yeah. Can you think about that one? Yeah. Yes. Go ahead. Can someone pass a mic to her? Yeah.
Don't switch up this pipe, please. Don't switch up the light. Don't switch up this.
Yeah.
Sir, when the formation of SS bonds occur, the molecules become big, so that when it goes to the SDS page, there will be a change in the molecular weight difference, the change in the size of the proteins basically. So that will affect the process.
Yeah, the mobility of the
Mobility of
proteins.
the
Yeah.
protein.
So what she is telling that when you have a disulfide bond, right, the size of the molecule is big. Now imagine, forget about the exact molecular weight of this protein. I say that the molecular weight of this protein is 100 kilodalton, right? The entire thing is 100 kilodalton. So now if I run the gel, now imagine this is the marker, and the marker is 100 here. And where do you still get your protein? Next to it, right? You will get your protein here, 100 Dalton. How do I say there is a disulfide bond here?
No, sir. If there is more cross-linking occurs, at that time, 2-3 molecules interact and form a bigger molecule. That's what I was saying.
Now imagine there is an interaction between molecule and you get a bigger molecule, and that comes over here, imagine. How do I say it's a disulfide linked? You're right, but how do I say it's disulfide? There's a lot of other interactions can also do this job. You can get two, that we cannot visualize if you get 2 bands on the same position. It all looks like a single band on STS.
If we do the comparison with the later one, where we have used reducing agent and where we have not used the reducing agent.
Absolutely. You have to do experiment twice now, right? You do an experiment twice, and that combining your answer with her answer, and that will give us the more information. How do we do that one? Please note that these disulfide bonds are reducible. Okay, that means this is how the disulfide bond actually exists on a protein, right? If you use a reducing agent, it will break this disulfide bond and it will give you 2-thiol group like this one. Basically, the protein was like this and it becomes like this, right? Originally it was 100 kilodalton. Imagine this subunit is 25 kilodalton and this subunit is, let's say, 75 kilodalton. You get two units, one is 75, other one is 25, right? What do we do in reality is that we run STS page twice or in two different wells, right? One we will run without adding a reducing agent, negative, no reducing agent. The other one is with reducing agent treated sample. Without adding reducing agent, the sample will give a band here. If you treated the sample earlier with the reducing agent, where do you get? And the lower band, right? Yeah, one you will get a 25 and one you will get 75. You see, get nothing here, right? And this information is extremely crucial for protein scientists. That will tell us whether your protein is having disulfide cross-linking, because disulfide cross-linking is an important cross-linking that happens in food system to create aggregates, what she was telling. Protein can make a bigger, larger aggregate with the better functional properties depending on the disulfide linking, and that disulfide linking we can visualize nicely with this particular method. Is it clear? We have to run samples two times. One, first you do not reduce, just run directly. Second one, use a reducing agent, for example DTT, dithiotriol, we use that one, or mercaptoethanol. We can use these two reagents to reduce the protein and then run the reduced sample. If you see the band disappearance and formation of new band, which clearly indicates that you have a disulfide linkages in your protein. Okay. This is an example to show you here. They are running two gels here under reducing condition and non-reducing condition. You can see that they get high molecular weight stuff in the non-reducing condition, and this is broken into different pieces. Then you can clearly say that these proteins actually containing disulfide bonds because you get a lower molecular weight fragments. Is it clear? So just to give an example here, this is again the soy protein isolate. If you run it in the non-reducing condition and this is reducing condition, you get a different pattern. Basically, these higher molecular weight fractions, as soon as you add a reducing agent, they are broken and get the lower molecular weight fragments. It clearly shows that soy protein isolate has a lot of disulfide bonds. Okay, so that information you can get easily with STS page. So whenever you want to run STS page, always run in two ways, non-reducing and reducing. You will simply get an extra information. Any question? Yeah.
Sir, is there another method to identify proteins apart from going through literature? Suppose we get different bands, but we do not know which band what it indicates. But we only get this.
There is something called there's something called two-dimensional electrophoresis. You can further separate them. However, there is no direct method to say purely based on STS page, this is my protein. No, impossible. But there is a lot of combination techniques people often use. What we do is that, just to answer your question, we cut this band and take it to the LCMS. That will help us. Okay, in gel digestion we can do, and then we can do LCMS. Then you can say, okay, this is conglycinin because I get approximate molecular weight by STS page, accurate molecular weight by LCMS. So it's a complementary technique. And some people also use Western blotting, and then you can do is what is that called, antigen interaction and so on. You can get more information. So an STS page alone cannot give you the exact information. It can only give an approximate molecular weight. I can't say it is glycinin just by looking into the band. I need to go back to the literature and say.
But I was unable to find anything in literature related.
Then you are in trouble.
Yeah, which protein is it? we submit what this band indicates.
Absolutely.
There
That's
was
the common problem of all PhD students, right?
Yes.
So you don't know, literature also doesn't show, and then becomes research active, right? Then you need to somehow find that fraction, do the fractionation. I'll talk about the fractionation later, and bring it to the mass spectrometer or take it to some other technique, do the Western blotting. You need to somehow identify it. There is no straightforward way, to the best of my knowledge.
Sir, one.
Yeah, we can do size exclusion chromatography. I'll get back to that in the later slide.
Sir,
Yeah.
one
It's
more.
a complementary. As I said, no single technique will tell you all the information. It's always it should be complemented by other technique.
Question, sir, if we treat protein by different methods, then sometimes we get thicker bands in, suppose in one treatment, and in another section we get band at same molecular weight, but it is thickness is less.
Yeah.
What it indicates?
It indicates two things. One, the loss of protein will be the direct indication, but usually, it's a good question you asked. People often say that the thickness of the band intensity is directly proportional to the concentration of the protein, and there is a lot of research going on. Image analysis. You take the image of this one, find out the pixels on this band, you can do the quantitative information, so-called semi-quantification. Basically, the intensity change is directly informing you that the concentration is changing. However, the biggest challenge is that the intensity is also depending on how this protein band is interacting with your Kumasi dye. Usually, when I say this is the same protein lane, now I can't say that this is having less concentration than this one. It is a less intense, can you see? The intensity is weaker in this compared to this one. Often I can say that, oh, maybe I have more of B than this one, but that is not necessarily true, because maybe this one is interacting nicely with the Kumasi dye, you get a thick intensity. This is having high concentration, but it's not interacting nicely with Kumasi, it appears weaker. So don't believe STS page directly to compare between bands. However, you can compare two different gels, because the same proteins. Here if I get a weaker intensity, then I can say the same time, weaker intensity, I can say that, okay, maybe I'm losing the concentration. It's very difficult to compare these two based on the intensity. Did I answer your question? So don't be fooled with the STS page band intensity. That's the take home message.
There is one instrument on which we can utilize this SDS page gel densitometer.
Yes.
That determines the density of that particular band, and
yeah.
That directly correlates with the concentration of the protein.
Yes.
And many of the researchers have done this.
Yes.
So this is contradictory, means as
Yes,
per
I don't believe in that data. You can always use between the same band. Yes, you can compare. For example, same protein can be compared. Different proteins is difficult to compare. I mean, I am not sure about the instrument you are talking about, but the people do the pixel analysis, the image analysis basically, and that is something you should keep in mind, that the interaction between Kumasi dye and the protein, that varies a lot. For example, we know that the nepin doesn't stain nicely with Kumasi, although you have a lot of very high concentration. So you can't compare nepin with cruciferin concentration. It's a bad idea. Then you have to go for SEC or some other size exclusion chromatography or some other technique. And also it's something depend on the loading volume. If you do a small mistake here with the loading concentration, you mess up here. If your protein is aggregating, something is trapped here, can you see? Everything doesn't come down, then again you have a problem. So you have to be very careful with STS. I personally use STS only for the qualitative information. I don't believe with anybody say with the quantity with STS. I personally don't believe it. Some people do that one if they are confident with what they are doing, but I was never confident with the quantitative analysis with STS page. I don't believe it myself. There are some papers show that you can do it, but be careful. That's it. That's my message. Yeah.
I also did this SDS page during my PhD research work, and I found that as per the literature, to prepare the sample, you just have to add 2 milligram of the sample. I added that, but there was no band. Then again, I increased the protein concentration to 5 gram, no band, 10 gram, little band, and that too merged one. Then I increased the concentration till 20 milligram. And then the bands were very clearly, beautifully elaborated and separated as per the SDS page. And the voltage, I fixed the voltage, but I vary the ampere. current.
Yeah.
So on the basis of that also you get the very beautiful separation of the,
Yes.
the protein bands.
Yes, yes.
That also defines.
Yes, yeah, absolutely. So it's basically very simply because the type of the gel you use. It's very important that the percentage of the polyacrylamide what you have it on the gel, that also determines the mobility of these bands, and also the voltage or the otherwise current here actually the real thing that moves the protein. But one thing I can always tell you that the order they elute is not actually compromised, even if you use it in different labs. It's always elute on the same order, and that's the reason why you always have to run the molecular weight marker next to the proteins. That will help you. But in terms of the loading volume and the loading weight, it's all dependent on the type of gel you use and the type of staining you use and the concentration of the staining agent. All these things matter. It's very difficult to give a straightforward answer. Often every lab will have their own STS page running method. It's very common, right? I've been working in so many labs. My lab has a particular way of doing STS page. Other lab has a different way of doing it because it works. They know what is the glitch here, and it varies actually. Yeah, yeah.
That the percentage of gel you are preparing,
Yeah.
like we prepare two types of gel,
Yeah,
stacking gel in which the samples are being loaded and the running gel.
Yeah, yeah.
So the concentration of the running gel is very much important in the clear illustration of the protein bands you want.
Absolutely. Yeah, yeah.
And if you want the higher molecular band, you have to go with the lower one.
Yeah,
right.
yeah, yeah.
And if you really want the lower one band, then again you have to go with the different concentration of the running gel
Absolutely, yeah.
percentage.
Usually the most of the STS page, let's say this is the maximum is 200 kilodalton or something like that. Often the cutoff is like around 3000 dalton or 3 kilodalton, and below that STS becomes tricky. Like you can get a faint band around 3, 3.5, 4, that's a max, and below that one you don't believe in that one. It's compromised. Okay, this is a disadvantage of STS page also. You can't get a very low, you can't separate amino acid with STS page. We're talking about 200 dalton. Yeah.
Molecular proteins are being separated with the help of acid electrophoresis.
Yes, exactly. Yes. It's again another technique and another problem. As I said, you need to have a combination of different techniques to get the information that you want. No one technique gives you one information. That's a good technique to use. Is it okay? Clear? Yes? There is a question? Yeah, the last bench, yeah. Okay, what do I have next? Yeah.
Sir, when we do STS page, what does it mean if we get a smear instead of bands?
Good question. That's one of the always biggest challenges with plant-based protein world. No STS page looks so beautiful. It's like always a smear. The smear, again coming back to my very first slide, the first slide I was talking about plant proteins, how complex they are. I'll try to draw it in here. So you have a plant protein, it's a powder, right? And as I said, this powder is mixture of albumins, globulins and prolamines, right? Again, they are mixture of different subunits, right? Again, they are mixture of different isoforms. So basically, it's mixture of too many things. When it comes to isoform level information, there is a very small difference in their mobility. And if you start doing STS page of that one, you don't get a clear bands like you get it for the milk protein. They look super beautiful. You get a smear for the proteins in case of plant proteins because it's a mixture of different proteins with a small difference. And the STS actually page cannot separate that small difference very nicely as an independent band. Especially if the protein is aggregated, you get a smear. It indicates that you have a hell lot of different types of proteins. You can't use STS page. That's the reason why if you do the reducing agent, they become a little bit much more beautiful now. Yeah.
In my case, if I add a reducing agent, will it give me a?
Yeah. In 90, there is a 50% chance that you get a better beautiful bands if you add a reducing agent, because now the protein will separate much better because there's a huge molecular weight difference. So.
But
yeah.
I have already added with reducing agent.
No use. You did not get any info.
No.
Then you have to use another technique. Come for the size exclusion chromatography now, or Ashim. Yeah. Yeah, yeah,
which is loaded in the stacking gel,
yeah,
you will get the smear one,
yeah,
the first one.
yeah.
And but when you reduce the concentration of the protein to an appropriate level, then you will get the clear bend.
Bands like this. It's not always the case. There is a 50% chance it's a concentration dependent, and I mean, I fully agree with you that you play with the concentration first, reduce the concentration, and see you get a nice band. But it's not always the case. You can see here also there's a big smear here, right?
Yeah.
It's the smearing intensity will reduce when you reduce the concentration basically. And some bands will appear as a nice one because then you have the relative concentration will change, and you might get some bands as a nice band, but you always have this faint smear in the background. So play with the concentration. Good point. Yeah.
Okay, sir. One more.
Yeah. 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 Go ahead.
Add on to the experience of the other people, my experience was I did 1 year.
1 year.
Yes, yes,
Yeah.
get my desired result.
Yes. Yes.
So I did so many mistakes and I learned from them, and I have documented that also.
Yeah. Yeah.
I also can add on these.
Yeah, yeah, you play around with, yeah, spend 1 year and to get a nice band. Yes, yes, there's a lot of options, trial and error you can do to get the one you want. But often what we generally do, at least in our laboratory case, we try 2-3 times, it doesn't work, forget about STS page, let's go for size exclusion chromatography. So we do that. So yeah.
Sir, and one more thing I got to know from a life science professor. He
Yeah.
asked me at what pH do you extract protein. I told at 12 I extract,
Yeah.
and I precipitate at 3.6.
Yeah,
So,
yeah.
so he said that protein will be denatured, so that's why you're not getting bends. Is that true?
No, that's not necessarily true. That's not necessarily true. Because before, if you do the native, there's another called native gel. I'm explaining only the STS gel here. There's a native protein gel can also be done, and there this could be a problem. But in case of this one, if the protein precipitates here, for example, in this case, imagine if protein is precipitating, it's your duty to bring every protein into STS tube. You add STS directly to this one. If you're only taking the supernatant to the STS page, you're actually losing already something. Then that might be a problem. But it's always recommended that the protein is precipitated, you add STS, it will become dissolved, and then you bring it to the STS page gel.
No, sir. What he said is the extraction conditions you did.
No. That's unlikely to happen. Because
yeah.
unlikely to happen because as long as you bring all the protein to the STS page, it should appear, because STS will, by default, it will denature the protein.
I have did multiple times myself, and I have given outsourcing also, but I got the same smear.
Yes, this is something problem with the protein. I don't say that this is another disadvantage of the STS page. It's not the universal method that will help you separate everything. No, STS runs really well with milk and meat proteins, beautiful. Plant proteins smearing is a nightmare. It's always there. So trust me.
Thank
so
you.
I only show you the best results here on the slide. Huh? So just to make your life things look easier. But in reality, I know what you mean. Yeah. All right. So remember this slide last time, protein polyphenol interaction. Polyphenols can interact with proteins and can damage the protein or improve the solubility, and those things can be visualized. I'm going to show you another technique now, which is complementary to the STS page. Now we have a protein here, okay, and the polyphenol is attached to the protein, right? And remember, polyphenols will always have a functional group, OH. Now I'm talking about protein polyphenol covalent bonds. Please note, it's not a non-covalent interaction, it's a covalent interaction, right? Now I have a two protein, one attached with polyphenol, one without polyphenol. Can you see? This is with polyphenol and without polyphenol, right? I run STS page of them. I don't have a real experiment with the plant proteins, but this experiment we have done with the milk proteins. I'll show you how you can use this technique. This is my own result. We took the milk and we added green tea extract. You know what is green tea extract? Which contains a lot of polyphenols, epicatechin, epigallocatechin, and all these catechins you have it, right? And we also know that the milk proteins interact with the polyphenols nicely and make a covalent adduct, okay? Then we ran through the STS and you get a separation. Now this bands actually contains protein plus polyphenol together attached. Anything that is non-attached will run away because the separation takes place only for the proteins in STS page. All the free polyphenols which is not bonded will vanish, which will just come to the buffer here. Do you agree? Now the bands what you see, it contains protein plus polyphenols. Now if I take this one and put it into the Kumasi staining, what do I see? I see exactly the blue bands, right? But can I differentiate polyphenols there? I just see again the same protein band, but I really want to see whether protein is now attached to the or bonded to the polyphenols. How do we do that? This is a technique we use in our laboratory. We use a technique called Western blotting. It's a common technique in molecular biology. What do we do in the Western blotting? We take the STS gel and we put the nitrocellulose paper or what is it? We also use PVDF, right? Nitrocellulose or PVDF paper, and we transfer all the band to the paper now, right? That's actually a blotting instrument is there. It will quantitatively transfer the bands into the paper now, right? Now you get a Western blotting. I hope I have a picture here. We get a paper like this. Now all the bands are on the paper, right? This is called nitrocellulose paper, nitrocellulose paper, right? And now this paper, now quantitatively transferring all the bands here, now this bands contains again protein attached to polyphenols. And then we put this paper, we spray a reagent called NBT. NBT is nitro blue tetrazoleum, okay? Nitro blue tetrazoleum, which is actually a redox reagent. It likes to interact with the polyphenols. Now the polyphenol attached on the protein contain hydroxylic group. It's an antioxidant. Polyphenols are antioxidant because it has OH group. Now this will interact with these polyphenols and it will be reduced to form a formazan, and that color is actually purple. Now if you see a purple band on the protein, it indicates that protein is attached to the polyphenol. And this is a fantastic easy technique to use to understand protein polyphenol interaction. You are welcome to use it. Is it clear? It is a complementary technique to STS page. STS page will only give information about protein, nothing else. Now I want to see the protein polyphenol interaction. What do? I transfer all the protein bands into the nitrocellulose paper, and then I stain it with a reagent called NBT. NBT will be reduced to formazan. The formazan is actually having the color purple, and if I see the purple color band, which clearly indicates I have a protein polyphenol interaction. Is it clear? But one of the disadvantages is that if your protein is glycated, Maillard reaction, they also give a purple color. Be careful. Okay, I'm not telling it selective to polyphenol, right? That's one of the disadvantage here. Is it clear? Any question here? Any question? No. We take a 10 minutes break. I know I'm supposed to finish my lecture now, but I have another lecture to cover, but 10 minutes break, okay? We'll come back. 10 minutes break? Yeah. All right, so welcome back. So from the break, we have been discussing about different techniques to characterize proteins. We just heard about ST space and the technique that can be used to understand the composition of protein, especially the apparent approximate molecular weight of different proteins in alternative proteins. Is not only used for the alternative protein world, proteins in general, biochemistry and also in food science. Is it okay? All right, so now I would like to little bit move on, but I have to make an announcement here is that this class will take another 15 minutes now, and then I'll stop, and then we'll go for a lunch, and we'll be back at 2 o'clock, okay? And the remaining portions of chapter number 5, the lecture number 5 and the lecture number 6 I'll take in the afternoon. Is it okay? Excellent. So 15 minutes more, I'll see how much I can cover. Okay, we are on the wrong slide. Okay. Okay, we have a chromatography, and I'm going to show you how chromatography can be used to characterize proteins. How many of you heard about chromatography? Super nice. How many of you done chromatography? Okay, just take a mic and just, in order to take a mic, just mention what are the types of chromatography you have done. Paper chromatography, I'll just write the word paper chromatography. Thin layer chromatography, what we call TLC. Yes, and then HPLC, what is the full form?
High performance liquid chromatography.
High performance liquid chromatography, HPLC.
Size exclusion chromatography.
Gas chromatography, size exclusion chromatography, SEC. Anything else? LCMS is a liquid chromatography combined with the mass spectrometry. Anything other chromatography? Column chromatography, CC. There's a lot of branches. Anything else? Affinity chromatography, gel filtration chromatography, HPLC again, highly chromatography, high interaction chromatography. It's a lot of different chromatographic techniques. I just want to tell you that you already know many things about chromatography. Now, let's discuss about what we mean by chromatography and how the word chromatography come. Now, can somebody define what is chromatography? Can somebody try to define what is chromatography? Separation based on color. That's a beautiful definition to have, because if you look into this word, chroma means color, graphy means graphs or writing, right? It's basically color graph is writing. Separation based on the color. Now I'm asking this question. Is everything colored in the world? No. Are proteins colored? No. Are amino acids colored? No, right? It's actually original definition, because back in the days they tried to separate things based on the color. Especially there was a guy, his name is M.S. Twist. Right? He is, I think, he was Russian, Italian or Russian Ukrainian scientist, if I'm not wrong. Around 125 years ago, he was there, and what did he do was that he took calcium carbonate, the chalk that we write on the board, right? And he made a column out of calcium carbonate, and he really wanted to separate what exactly present in these leaves, for example. You know, leaves is green in color, right? But it's not just green in color, right? For normal person, leaves are green. For scientists, leaves are green, but it's also green plus something, plus, right? And he really wanted to see what are the rest of the things present in this one. What did he do? He crushed these leaves and put it into a solvent, I think, if I'm not, ethyl acetate or something like that. He used a solvent, and then he loaded it into this small column, the calcium carbonate column, like this one, right? Then he poured more and he loaded the sample. This is the extract of leaf. Then he added more and more solvent. Then he visually could see that the separation of colored bands. Then he realized, okay, it's not just green, it's actually the extract contains too many other components. He could see orange color, he could see green color, he could see yellowish color, and lot of different colors he could see. Then we later realized that is nothing but different components present in the leaf, right? We know that carotenes, carrot, orange in color, right? We know chlorophyll, green in color. We know xanthophylls, yellow in color, right? Basically, the leaf is mixture of different photosynthetic stuffs like chlorophylls, A, B, xanthophylls and orange. And for naked eye in the mixture, they look just as a green, but you can separate them in individual components, and then that becomes chromatography, separation purely based on color. Then this was started in 1903, right? Now it is 2025. Thousands of experiments have been done. Numerous different things have been separated, and numerous detectors are attached to the chromatography, because our eye is the best detector in the world, right? We can do the eye test now. You can see the yellow color I can see, but our eye is not every time is right ones to see everything, right? Colorless compounds we can't see. Then we started attaching numerous detectors to see the unknown things that we cannot see in our naked eyes, and that becomes more complicated field. We call it as chromatography world. So I'm going to teach you a couple of chromatographic stuff, mainly thin layer chromatography. I don't teach you paper chromatography today at the interest of time, a little bit about column chromatography, and mainly about HPLC. Okay. I think we can start with some fundamentals of chromatography, right? Whenever you take a chromatography, we have to remember about three different words. One is stationary phase, the second one is mobile phase, and third one is analyte. Stationary phase, as the word says, is static, doesn't move, right? Stationary phase. Now, in his previous experiment, calcium carbonate could be considered as a stationary phase. What is mobile phase? Exactly, the one that moves through the stationary phase is called mobile phase. Mobile phase can be liquid, can also be gas. In the gas chromatography, right? The mobile phase can be liquid as well as gas. If it is liquid, it becomes a liquid chromatography. It becomes gas liquid chromatography. Okay. Is it clear? And the third one is called analyte. What is analyte? The solute particle that we want to separate, it's called analyte. I am going to use these three words continuously in the future classes. When I say stationary phase, it's something doesn't move. When I say mobile phase, it's something moves, and the analyte is something that we want to separate. I don't define it again and again. Is it okay? Cool. So, as I said, the chromatography based on that one is divided into gas, liquid, thin layer and paper chromatography, and the column chromatography. They all come under the liquid chromatography category, and then HPLC. It's high performance liquid chromatography. Now I'll start with thin layer chromatography. In the next 5 minutes I'll complete this one. What have you done thin layer chromatography in your life? Yes or no? It's a very basic experiment they teach in high school or in colleges basically, if you've taken chemistry classes. You have not done? Have you done paper chromatography? Paper you have done, right? It's a complementary to paper chromatography, I can say that one. What do we have here is that the stationary phase in thin layer chromatography often is silica. Okay, it's not necessarily always silica, but often it is silica. Please remember that silica is polar in nature. Silica is polar in nature, right? The mobile phase is organic solvent. It could be ethyl acetate, it could be hexane, and lot of other organic solvents they use, right? This is usually relatively non-polar, right? One thing keep in mind in chromatography, polar likes polar, polar likes polar, non-polar likes non-polar, polar hates non-polar, non-polar hates, keep that in mind. Don't get confused with positive and negative. Positive loves negative, negative loves positive, polar loves polar, non-polar loves non-polar. This is the interaction, you have to keep it in mind in chromatography. Okay. Now, what they do here is that they take a chamber, it's a glass jar basically, and they take the solvent that you want, mobile phase, right, and then take the thin layer paper, like this is a thin layer paper, and we just make a mark here like this, right, and we make a drop of your sample, one drop, right, one drop of sample is placed here, and then it is put inside the chamber like this. You should not touch the drop. Now what happens? The solvent moves against the gravity because of the capillary action. The solvent moves above the against the gravity. When it is moving, it will start solubilizing the analyte, right? When it is moving in this direction, it will start solubilizing the analyte. Now, this analyte, for example, is mixture of 3 different components, right? It contains yellow component, red component and the blue component, right. Now, the solvent is moving in this direction. Initially, it will start dissolving this component, like, and slowly the solvent moves in this direction, right. Who will move faster in this case?
Degree of affinity for the solvent.
The one having the lower degree to the solvent or the lower degree affinity to the stationary phase? Okay, let us not confuse each other. In this figure, who is moving faster?
That is having more solubility to the mobile phase.
Yes, I mean just look into the just figure. I will get back to this point. Let us talk about color and things here. Who is moving faster?
Yellow.
How many of you think it's yellow is moving faster? I don't think yellow is moving faster. Who is moving faster? Look at the gel, look the paper properly, the thin layer. Blue is moving faster? No. Look carefully. I know it's a lunch, you are hungry, but look carefully. Who is moving faster? Red? No.
Solvent is moving.
Solvent is moving faster. Can you see? The solvent is over here.
Yeah.
The solvent is moving faster because solvent is non-polar. It does not like the stationary phase. It just want to get rid of stationary phase, so it's moving faster. When it is moving faster, it will take its friends. Can you come with me? And these are the analytes, right? Now, the yellow one does not like stationary phase as much as possible, so it loves the mobile phase. So, it moves along with the mobile phase. The red one, 50% it loves the stationary phase, 50% it loves the mobile phase, it moves slowly. The blue one, it really wants to stick to the stationary phase, it hates mobile phase is pulling it, it says I do not want to come. Based on this mobility, you get a separation, that is it. That is a thin layer chromatography, purely based on the love for the stationary phase and love for the mobile phase, the interaction, the affinity. Clear? Whichever likes the one, it stays with that. If the analyte loves the mobile phase, it goes with the mobile phase. If the analyte loves the stationary phase, it tries to stick to the stationary phase. But there is a difference for this love, and that makes the separation. Is it clear? It's a fundamental of chromatography. The same knowledge is applied for paper chromatography, HPLC, many things. It's just about the affinity. Is it clear? Now I have a small, no, we stop it there now. It's already 12:45. We take the lunch break now, and we will come back at 2 o'clock. Okay? So we take a lunch break. Yes, welcome back to the afternoon session. So, lecture number 5, analytical techniques for protein characterization, chromatography and spectroscopy, and we have covered almost all spectroscopy in the morning session, and we have started with chromatography, and we began with thin layer chromatography just to showcase you how exactly the chromatography works. We heard that polar likes polar, non-polar likes non-polar, and opposite does not exist. So, polar hates non-polar, non-polar hates polar. And in a chromatographic system, there is always a stationary phase that is stationary, does not move, and there is a mobile phase that moves over the stationary phase carrying the analytes. Analytes are nothing but the ones we want to detect, separate, whatever, right. These are things we have realized in the last session. So, now I have another TLC plate just to check your knowledge. So, I have a TLC plates with the dot here, can you see? And this dot contains 2 types of amino acids now. The dot is a sample now, okay. 2 types of amino acids I have. One is leucine, other one is glutamic acid, glutamate. Leucine is a non-polar molecule and glutamine is a polar molecule, right. I put this plate into a TLC chamber. Please note that the TLC plate is polar in nature, and then I started doing TLC of that sample. I got 2 components, I show it as red and green now. Now, tell me whether the red is leucine or glutamic acid. How many how many of you think red is glutamic acid? How many of you think red is leucine? We still have a 50-50 chance here, now I am confused. Okay, could you please explain why do you think red is leucine?
Because the mobile phase is non-polar and this leucine is also non-polar, that's why.
So, he thinks the mobile phase is non-polar. So, glutamic acid is
Polar.
polar. So, should it go together with mobile phase or it should stay on the stationary phase?
It should stay with the stationary phase.
So, now should we change the answer? So, which one, red is glutamic acid or leucine? The red is glutamic acid. I am showing it's leucine. So, glutamic acid is polar, stationary phase is polar, polar likes polar. Now, glutamic acid is polar, stationary phase is polar. Where did it wants to stay? With the stationary phase, it does not want to move together with the mobile phase. Leucine is non-polar, mobile phase is non-polar, leucine likes non-polar mobile phase, so it goes with that. It is a very important exercise to keep it in brain. It often confuses you. If you do not understand this basics of chromatography, your life will be tough in chromatography. I am telling you. That is the reason why I have started with thin layer chromatography, not directly with HPLC, just so that you need to understand this concept. Is it clear now? Cool. So, just to give an example, you do not have to remember this one. Thin layer chromatography can be used to identify different amino acids, but nobody uses thin layer chromatography in protein world. Can you tell me why? We can't use thin layer chromatography for protein analysis. We can, but it is almost impossible. What could be the reason? Yeah.
The proteins we deal in real life situation is a crude protein,
Yeah.
and so it will be tough to separate using TLC.
One of the things is that TLC is not so efficient in separating. It works with some of the organic, even organic chemistry people always use TLC to check the purity, but in protein science, especially in food science, we are talking about too many different things, right. We have proteins, we have some other things, then it is a problem. Any other disadvantage? Why can't we use proteins?
Protein is a complex system.
Protein is a complex, plus protein is heavier, right. So, amino acid is a small tiny one, it can move along with the software, along with the solvent, but the protein is a heavier molecule, and the capillary action perhaps is not sufficient enough to take that into the higher level, right? That is difficult to drag the proteins. You might get a small smear, then that's it. The third problem is that you need to see, TLC always need a detecting agent. Either you can use a UV light or you need to spray something to see the color, right? And it's always challenging with protein sometime. Make sense? So TLC, don't try to use it for the protein science, but I used TLC just to give you an examples of how chromatography works. Does that make sense? All right. So now the one we are going to use in food systems or in protein science is usually column chromatography. Usually column chromatography is basically the TLC knowledge can be translated to column chromatography. It works with exactly the same phenomenon, but in an opposite way. In TLC, we have a plate and we have a sample and we put a inside the solvent and things move in this direction, right? In column chromatography, you have a column and the stationary phase is filled in the column and the sample is loaded on the column, and then you load more and more solvent and the separation happens in this direction through the gravity. Sometimes you can also apply pressure and vacuum, but usually it's a gravity separation. Make sense? So in TLC things go in the up direction. In column chromatography it's towards the gravity. That's the important difference. And this is how the column chromatography looks like if you want to separate some colored component from columns, just to give you an idea, right? And these columns can be made small, big, whatever size you need. And there is a lot of different types of stationary phases are available for separation and purification. Okay? All right. Now, I'm going to talk a little bit about how we are using this column chromatography to separate and purify proteins. Please note that chromatography is used not only for the identification and quantification process, it's also used to get high pure proteins. For example, if you want to get more than 90%, in addition to extraction, in addition to fractionation, in addition to alkali extraction, in addition to sometime dialysis, you have to use column chromatography. You can reach 95, 97, 98, 99% purity also sometime. You know that you can buy some milk proteins of 99% purity, right? Those can be usually purified. Of course, there are a lot of other technology. You can use column chromatography to get that 99% purity. That's the advantage here. Okay. Now, this is just nice illustration I got, and you can see that imagine these are your proteins and you have a lot of impurities here. These impurities in our plant-based world, it could be polyphenols, it could be anti-nutrients, it could be small peptide, it could be minerals, whatever, right? But we want only proteins separated, right? Imagine I just wanted to separate the green color protein here. How do we do that? And there is a lot of options. Let me take one by one. In general, column chromatography is nothing but you have a column and filled with some beads or some powders, and that is what we call stationary phase. Different numerous 100 types of different types of stationary phases are commercially available, and that is called stationary phase. And you put your sample in this range, in particular region, and then the mobile phase usually solvent. You can use different types of solvent here: polar, non-polar, aqueous, salt solution, alkali solution. Different kinds of solutions you can use as a mobile phase, depending on what you want to separate. Okay. Now, I will just show you one of the most important techniques what we use today to separate proteins and purify proteins is called size exclusion chromatography. Sometimes it is also referred to as gel filtration chromatography. Okay. Size exclusion chromatography, and I often say it as SEC or SCC, SEC. Okay. Size exclusion chromatography. As the word says, it is something, the separation principle is something related to size. Now, please listen to me carefully. It is an important chromatographic technique you have to learn. In size exclusion chromatography, we have a column and filled with a special type of beads, right? It is filled with a special type of beads. I will just try to illustrate here. This is the column, and the columns are filled with special types of beads like this. Okay. And these beads are porous, means the beads are spherical, but they have holes inside. They are all porous. They are all porous like this one, okay. Now I want to separate rapeseed protein isolate, for example, okay. Rapeseed has two kinds of protein, one is called cruciferin, its molecular weight is let us say 300 kilodalton, and we have another protein called nepin, and whose molecular weight is let us say 14 kilodalton, and it has lot of polyphenols, okay. Polyphenols having the molecular weight let us say 300 Dalton, these are all other kilodalton. Can you recognize there is a difference in the size here? The molecular weights are different, right. Now I want to separate them, these three separately. Now let us see how I can do this one. I have this matrix here, and that is a solution containing protein and polyphenol, this is the matrix, alright. I take it and I pour it on the column like this one, alright. Now I will add some solvent and etc. things. Now the sample is here, right. The sample contains cruciferin, I show it as a big bead here, big one, and the second this one is nepin, and we have a tiny one is let us say polyphenol, everything is here now, right. Then I have a small this one, I am collecting the sample, okay, this is me, right. Now I want to collect the fractions. Now I load. Now tell me which fraction I will get the first. So polyphenols, how many think that we will get polyphenols first? Okay, we have polyphenols first, and any other answer? Yeah, I have to make one thing clear, size exclusion chromatography, forget about the interaction for now, okay. Forget about the interaction, look into the diagram, okay. Think about, forget about that polar interaction I am telling, right? Not the interaction in general. Think about which one comes first.
Smaller?
The polyphenols, how many, the smallest one comes first, how many you think? Okay, why do you think the smallest one comes first? Could you pass the mic to her? It could be a right answer, I am not criticizing you. Yeah. Yeah.
As small as the particle is, it moves faster.
Okay, the small particles moves faster, right? I don't know. So small particles moves faster, and she is telling that polyphenols is smallest in our case, and she will collect, this is her, she will collect polyphenols first. How many of you disagree with her? We have a disagreement there. Could you please pass a mic to him and let us see why he is disagreeing with you?
So if the size of polyphenols are such that they get stuck in the pores of the beads,
okay,
so they may get stuck there, and the nepin then would move fast.
So he thinks nepin will come. Now we have another gentleman here. He thinks the first one is nothing but nepin. Why? Because he thinks the polyphenols will be stuck here, and nepin comes, so he thinks nepin. Oh, there is an answer here. Come on. There is a nepin. Yes. Who is left now? Cruciferin. Is there any friend of cruciferin? No, I am a friend of cruciferin. I think cruciferin comes first. Who will agree with me? She agrees with me. Let us see why.
Because of the gravity, it will come fast.
She thinks cruciferin comes fast because bulkier, and the gravity will make cruciferin comes first. That is her understanding. Oh my God, now I am confused. So everybody is giving different answers. So what is the right answer? Now listen to me carefully, okay. We have the column filled with the beads. Beads are spherical, but each bead are porous in nature. They have small holes, porous in nature. Now you pour a solution containing different proteins of different size and a polyphenol with the smallest size in this case, for example, right. Now the smallest polyphenol, what it will try to do is that it wants to come fast. I agree with that, smallest, and it wants to come fast, but it cannot come fast. Why? Because these beads are tightly packed. Instead of coming fast, it will pass through the pores. Okay, it will pass through the pores layer by layer by layer by layer by layer, individual pores, 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 right? It takes forever. The last one to come is polyphenol, the smallest one, and the nepin comes the second one because it wants to come in the porous, but there is also a gap between the beads, right? And the second one is coming the nepin. The last, the first one to come is the biggest one, is cruciferin. Why? Because cruciferin is the largest size. It wants to go inside the pore, but it can't go because the pore size is so small. Then you are still pouring the solvent on top of it. It tries to come in between the beads, right? It's the only case the larger one runs faster in size exclusion chromatography. Keep that in mind. Right, it runs faster, so the cruciferin comes first. That is size exclusion chromatography. The bigger one comes first, and the smallest one comes at the end. The biggest one comes first, and the smallest comes at the end. Is it clear now? So our first fraction will be cruciferin, our second fraction will be nepin, and our third fraction will be polyphenol, and this is how you can actually separate multiple things using size exclusion chromatography. It can be made as a small column, an open column. It can also be made as a column for connecting to the HPLC. I will talk about HPLC later. So, you can make it in a different dimensions, and there is no interactions here to play, to be frank. No polar, those are very, very minor because we always try to use an inert bead. Okay, it's purely because of size. 99% I would say, maybe there is small interaction for some other reason, but it is purely because of size. Is it clear? So, in size exclusion chromatography, you can see it clearly that the bigger particle cannot pass through, it stays away and passes through the bead, between the bead, and it comes first. No doubt? So, now you can explain size exclusion chromatography to anybody, right? Do not forget this. Then we have an, there is a question? Yeah.
Does density play a role?
Density of what?
Like the macromolecule for means suppose size is same but suppose small size but higher mass then.
Yeah, I mean basically yes or no, because if you are using the column chromatography, an open column, the separation is purely based on gravity. That you know, the gravity is directly proportional to weight and so on. That's true. Maybe I am wrong here, but in case of column chromatography that we use in HPLC, we always put a pressure on the left hand side. The solvent is pumped at high pressure, and basically then usually the size is the main governing factor. Yeah, I am not very sure the density will play a role. Density is what is the formula? Density
mass by volume.
Mass by volume, right?
Volume again is correlated with the size.
Yeah, the volume is related to the size. Yeah, it might play a role. You're right. Yeah, agree with you. Yeah, of course it's the same thing, right? If you have a bigger volume, then now density is inversely proportional to the size. So makes sense. Yeah, agree with you. Any other question? Okay, I have to quickly move on now. So we have another chromatography called affinity chromatography. It's also used quite often in molecular biology for the purification of certain specialized proteins. In food science, we can use it for purification of some allergens, for example. In this case, you also have a column, but this speciality of this column is that the beads containing some sort of ligands. Can you see? The beads contains some types of ligand, L, right? Now, if you add a protein, this is my protein, right? If the protein likes the ligand, it attaches with the stationary phase. Whatever that does not like the ligand, it just passes through. So, it is a two-step process. You add the sample. If the protein likes the ligand on the stationary phase, it will just attach on the stationary phase, and the remaining things come out first, all right? But we want this protein, can you see? To get this protein out, we add an elution buffer. It could be a salt solution, it could be another solution with high pH and so on, and then the interaction between the stationary phase and the protein will be broken and the protein comes in the second step. Is it clear? It is a very simple way of doing things, but there is a lot of different types of ligands that are available, but the basic principle is the same. You have a stationary phase with the ligand like this, okay? Stationary phase with the ligand, all right, and this is my protein, right, and I also have a lot of unwanted compounds, right, and I pour it on the ligand now, and the ligand likes the protein, so the ligand will attach to the protein. Other things will come down first, that is this step, right. Now I actually want my protein also back at the end of the day. To remove the protein, I add another solvent, and the interaction between ligand and the protein is removed, and the ligand comes down in the second step. So this is called affinity purification. There is an affinity, is a love between ligand and the protein. Does that make sense? Okay. One of the beauty of column chromatography is that they can be made as big as this room also if you want. They can be upscaled to a crazy scale, provided you can control solvent and other things. Basically, you can see now this is one figure I got it from the internet. The person is standing, you can see that the size of this column chromatograph, it could be of this size, it could be of this size, and so on. And sometimes the ligand what we use there, the stationary phase, that you can reuse it. It's not that it's always use and throw. So you can reuse it several times, so you can make a high quality purification on a cost effective way. But I don't say it will come forever, but can be used for several cycles depending on the impurities present. Any question? Is it clear? All right. Now I will move on to the most important thing, is called high performance liquid chromatography, HPLC. How many of you heard about this word? How many of you used HPLC? How many of you have seen HPLC? Super. So there is some basic knowledge, but I will not go very deep into HPLC things here. I can teach it for 3 hours, to be frank, but I can, since the time is a limiting factor here, I will try to show you the most important things only. Is it okay? All right. HPLC means high pressure liquid chromatography or high performance liquid chromatography. So in the column chromatography, we have columns with beads and we load here, and the separation is usually based on the gravity. So I collect the fractions, and who is collecting the fraction? Let us say it is a PhD student, okay, and the person has to wait until every fraction comes. Do you agree? And since it is a gravity separation, sometimes it takes forever. You know, sometimes you will get drop by drop, 1 drop per second, sometimes maybe 1 drop per minute also. Then to collect that fraction, basically he has to spend half of his PhD maybe in some cases, right. So that is a bottleneck, that is a problem with the column chromatograph. I don't say it is a bad one, it is actually a slow process. But if I can make it faster, I need to flow things faster in this direction. Do you agree? Right? How do we make it flow faster? You apply pressure, which direction? From top to bottom in this case. So you apply pressure in this direction, or you have to apply vacuum in this direction. Do you agree? Either you apply vacuum or you apply pressure on the top. So basically applying the pressure on the top is always easier, so that becomes a high pressure column chromatography. And basically then the column need not be vertical always. You can put columns like this, anywhere, any position you can put, because either side is kept, right. The stationary phase is inside and the solvent comes in this direction, and that is called column HPLC. It creates high pressure. Why? Because in this case the particle size can be reduced to very small. You know, when you reduce the particle size, you have more surface area. When you have more surface area, more interaction, more efficient separation. But the problem is, when you pack the small particles so tightly, the pressure increases. Do you agree? The solvent does not have a space to pass through now, because you have densely packed the packing material. So the pressure increases. You can get a very high pressure depending on the type of packing, length of the column, and the viscosity of the solvent. The pressure can rise 300 bar, 400 bar, 1000 bar, 1500 bar and so on. It's extremely high pressure I am talking about. To handle that pressure, to create that pressure, we need an equipment that is called HPLC, right. It's also called high pressure is simply because it handles so much pressure. It's also called high performance because now nothing is manual, things can be made more automated. So that's called high performance. Is it clear? Right, the column chromatography. Now I will there is a, have you heard about HPLC and UPLC or something? It just they are the same but different, right. Same mechanism of separation and everything. HPLC usually operate up to 400 bar. I might be wrong here because people have different claims for that one. UPLC can handle up to today's 1800 bar-ish, something like that. That is the only difference, right. You can even make a very fine particle based column with UPLC. Efficiency is very high, you can reduce sample separation time with UPLC compared to HPLC, okay. But if you have more questions, you can ask me later. I will not go into detail all these things. Just remember they are the same but different pressure operation, maximum pressure operation, okay. Now I will briefly show you the different parts on the HPLC, quickly show you. So HPLC has several parts, and the first one is the solvent reservoir, which is on the top of the instrument always. And basically in the normal column chromatography you have to pour solvents, right, manually, but here it is basically mostly automated. You whatever solvent you need, put it in the bottle, there is a pipe connection, it will do things most automatically, right. And then you have a most important part of the HPLC is called pump, because that is something handles high pressure and sends the flow in the right flow. If you want 200 microliter per minute, it will send exactly 200 microliter per second, and that is the job of the pump. It takes the solvents from the bottle upstairs, right, okay. And then we have a sample compartment. In the column chromatography, you remember we have a sample, it's one sample at a time, do it manually again and again. But even here in the auto sampler can handle, like let us say, depending on the type of the sampler, you can have 100 samples, 200 samples, 300 samples in an auto sampler, and it is also temperature controlled. You put it in the vials and go home, everything will be done automatically. It injects automatically, it analyzes automatically, everything is automated. You just have to place the sample in the right place. You do not do a manual injection, you do not do a manual loading. So it is having an automated injector and auto sampler with the temperature control, okay. And then the second important thing is here is the column, and the column comes with the different stationary phase type, which I will take it in the next couple of slides. And what actually happens is that you inject the sample here, right, and the sample goes through the column like this one, separation takes place, and then it comes to the detector. So HPLC can be connected to different types of detector. Remember, it could be UV detector, it could be fluorescence detector, it could be refractive index detector, it could be diode array detector is actually similar to UV, fluorescence detector I covered. It can be detected to the most famous MS, mass spectrometer, right. So LC is the plot where the separation takes place. Separation takes place here, and once the analyte separates, they come one by one, and that will be detected in the detector. Am I clear? And these are different types of detector depending on what you want to measure. If your analyte absorbs in the UV region, you take the UV detector. If your analyte is fluorescent, take fluorescence detector. If your analyte is having a refractive property, you take refractive index detector, for example, sugars, organic acids and so on. So depending on what you need, different techniques are available for the detection. Now I will talk little bit more about the HPLC column, okay. It is a very important subject. A HPLC column looks like this. Have you seen it before? It comes in a different length, dimensions and so on. An HPLC column look like this. It is usually made up of stainless steel. Can you tell me why they make it a stainless steel? Why not plastic? What's that? Yeah, one of the reason is corrosion resistant. Second reason? Third reason is second reason inert. The most important reason? To withstand pressure. I don't say plastic column does not exist, they do exist, but usually stainless steel, it's simply because we're talking about 300 bar, 400 bar, it's actually high pressure. If it's a weak plastic, it will just burst off. So that's the reason why you use a heavy metal, steel one as the column, and the stationary phase is actually inside this one. You can see the white color silica if you open these two nuts, but don't try that one at the lab, but because each column can cost you approximately 80,000 rupees to 1.1 lakh, 2 lakhs, something like that. Your supervisor will be super unhappy if you try to open it. You're welcome to do it, but don't tell that I told you to do that. All right, so how the separation takes place in HPLC column. So the mobile phase is moving in this direction. Okay, mobile phase is coming from the pump. Okay, from the solvent reservoir, mobile phase comes to the pump, and the mobile phase is going in this direction. Okay, and meanwhile you have injected the sample here. All right, this is the sample initially in the beginning of the column. Right, the sample contains lot of analytes. Let's say it has analyte A, analyte B, and analyte C. Right, and the solvent is moving in this direction, exactly like thin layer chromatography. Right, now separation takes place exactly on the same principle. If the analyte likes the stationary phase, it wants to stay with the stationary phase for a long duration. If the analyte likes the mobile phase, it wants to go with the mobile phase. So that is why you get the separation. So different analytes as a band. Can you see? Now tell me, among these three colors, which color likes the stationary phase? Blue likes the stationary phase. Now at the end of the day I get a detector. The detector gives you the chromatogram, which looks like this. Have you seen the chromatogram? Okay, this chromatogram is belonging to these three analytes. Now these three peaks in the chromatogram belongs to these three analytes. Now tell me, among these three peaks, which one is blue? Which one is blue? The last one is blue. How many of you agree? Do you agree the last one is blue? Yes, because the blue is retention time is longer, the retention time, and this could be the abundance or absorbance, whatever, right? So retention time is longer, it wants to stay in the stationary phase, it comes at the end of the day. So this is blue and this is red, and this is the first one is yellow. Clear? This is the basic principle of HPLC. And you can buy different types of stationary phase to separate different molecules. The stationary phase varies, and when the stationary phase varies, the mobile phase varies. Okay. Now let's move on to the same thing. Polar likes polar, non-polar likes non-polar, positive likes negative, negative likes positive. Remember that one. Okay. Based on that principle, we have different types of HPLC: normal phase, reverse phase, and depending on the size, you have a size exclusion chromatography, ion exchange chromatography, hydrophilic interaction liquid chromatography, high helic or helic. There are different types. I will try to cover, I have already covered size exclusion for you. I will try to cover these two, okay, quickly. In the normal phase chromatography, the column, this is the column, okay, this is the column. The column is filled with a powder, a very fine powder, and that powder is made up of silica, okay, very cheap actually, but it's high quality silica, it becomes expensive. So it is filled with silica, and this is the structure of that particular column powder, right? You can see that one. How does it look like? I would like to thank Waters here, because all the slides I borrowed from Waters, just to let you know. They had a nice fantastic presentation for me ready, and I just used them. So just to let you know. So it has a hydroxyl group, can you see? Okay, hydroxyl groups are polar in organic chemistry, you have studied, right? So the stationary phase is polar here, remember? Stationary phase is polar, right? This is how exactly the silica powder looks like. It's having a very fine particle with a high surface area, okay? Now, how the separation takes place in the normal phase chromatography. Now, I have a sample that contains 3 different analytes, right, and the mobile phase here we have to use non-polar. Can you see? The mobile phase we have to use non-polar. We are not using polar mobile phase. Stationary phase is what? Polar. The mobile phase is non-polar. For example, hexane. Now I have a question, why can't we use a polar mobile phase? How? Station. Exactly, she is telling that stationary phase is polar, mobile phase is also polar. Now the analyte is clueless with whom I have to go. Should I love stationary phase or should I love mobile phase? Now it's confused, huh? So that's not the life of this analyte. That is one of the reasons. That's a good reason. What is the second reason? Was it? Separation will not occur exactly for the same reason, that they don't want to move, they cannot move. Third reason? It does not elute out at all usually. Why? The biggest problem here is that if the mobile phase is polar, stationary phase is also polar, polar likes polar, stationary phase also likes mobile phase now. The now mobile phase wants to stay in the stationary phase and you are continuously pumping in this direction, but it does not want to go out. What do you create? The pressure will increase like anything. So that is not a good separation to do. Am I clear? That's one of, you need to have a kind of an opposite effect to elude them. Exactly what she mentioned. Somebody should carry somebody. If everybody stays it, that's a no separation. Is it clear? So now tell me, I have a 3 peaks now. Which one is blue? Huh? The first one is blue, right? This is blue. Now I ask you a question, which one is polar? Blue is polar or yellow is polar analyte? Yellow is polar analyte and the blue is? Wow, I'm so happy to get this answer. You understood chromatography. Thank you so much. So that's the right answer. Is it clear? Yes. Right. Now I have one simple question that I'll take only 1 or 2 minutes. You don't have to answer. Yeah. Why we don't use a normal phase chromatography for polar analytes, let's say sugar and polar free amino acids? Why can't we use it? We don't use it. We use reverse phase for that. Why? First is their polar compound, they really want to stick, I just don't want to come back because silica is so polar and it really wants to attack to the polar analyte and it's very difficult to remove it because your solvent is non-polar. Your solvent actually should like to a certain extent to carry them. It's not if they completely hate it, they don't want to carry it, right? That's one of the reasons. The second most is that, this is an important point I want to raise. If you go to any HPLC lab nowadays, you go to anybody, any collaborators, if you say no, normal phase, they say no, I don't want to run that one. We never use normal phase. Can you tell me why we are not using normal phase? No, time is not a big problem. As long as you know the analyte, no? Polar solvents are cheap, that's one of the reasons. It's not only because of the cheapness. Second one, availability. Third one, what is the problem with the non-polar solvents? Exactly, the non-polar solvents are very toxic. We need to use hexane, for example, in this case. And they are toxic, they are not environmental friendly, they are highly volatile. People know that if you breathe it, it's a problem. And plus, non-polar solvents melt so many things, you know, right? Yes. It melts plastic, it dissolves everything. So it's very difficult to handle this non-polar solvent in an analytical laboratory. So that's the reason why we don't use non-polar a lot nowadays. Only if in exceptional cases we use. Make sense? To avoid all these things, we have a reversed phase chromatography. As the word says, it's the reverse of normal phase. Okay? Reverse phase is just the reverse of normal phase. In this case, the stationary phase contains silica, but that hydroxyl group is blocked by a non-polar thing. Can you see? The hydroxyl groups are blocked by a non-polar thing. Have you heard about C18 column? You often hear that C18 reverse phase column, C18 reverse phase column is nothing but the carbon is total number of carbon is 18, right? You can see 1, 2, 3, 4, 5, 6, 7, 9, 10, 12, 13, 14, 16, 17, 18, right? So this is C18. That gives you the non-polar nature to the column. Please remember in C18 column or in the reverse phase column, the stationary phase is non-polar. Now let's move back again here. Now you will answer this question. In case of reverse phase chromatography, stationary phase is non-polar, mobile phase is polar, right? Now you tell me which one is the most polar analyte here? Yellow. Do you agree? Who will come first? Yellow will come first. Fantastic. It's just opposite of normal phase chromatography. That's it. Clear? I'm so happy that you're understanding reverse phase chromatography. There's a lot of variabilities we can use to maintain. I explained it in the most simplest way as possible, but it's a very complex system, HPLC. You need to play with too many things. You need to play with the column size, type of the mobile phase, the gradient program you're using, temperature of the column, what is the mobile phase you're using, and in which solvent your analyte should be dissolved. There's a lot of things you can change in HPLC chromatography, in HPLC, to get the best separation. Please note that for a chromatographer, we always love to get peak like this, a Gaussian, well separated peaks. This is the dream of a chromatographer. In reality, we get like this and too many problems we have when you start chromatography first. But when you optimize all these things, you can bring this to this one. Make sense? Peaks can be broad and peak can be tailing, peak can be fronting, the peaks could be split. A lot of problems you will see. By optimization, by changing all these things, you will get a nice chromatogram. Okay? Is it clear? This is something, the job of the chromatographer in your laboratory. Usually, if you go to any chemical laboratory or food analytical chemistry laboratory, there will be a dedicated person handling the HPLC. You have seen that one. They say, don't touch the machine. Oh, I don't want to give it to you. It's simply because it's not that they don't like you to train you. It has a lot of parameters. If you do a small mistake, things can go really wrong. For example, for HPLC column, they are tightly packed. If you forget to filter the samples properly, what it will happen? It will block the column. As I said, the column can cost you all the way to 1 lakh Indian rupees. And I don't want to throw that column, right? It's so expensive. Sometimes you put the guard column to prevent all these things. Still, the guard column is also expensive. So it's a lot of money to run an HPLC. HPLC is also a very, very nice equipment, but it's also very expensive to run. Just remember, okay? Compared to TLC. TLC could be 1 rupee, HPLC could be 1000 rupees, the price I'm telling about. All right. Now, I want to tell you one important thing. HPLC can be used for identification as well as also for quantification. Why? Because the area under the curve is directly proportional to concentration. The area is directly proportional to concentration. By injecting the known standards at different concentration, you can plot a calibration curve like you do it in the UV that you have done in a laboratory, right? You can make a calibration absorbance versus concentration, area versus concentration, perpendicular, and you get the concentration of the unknown compound. So, HPLC is often used for the quantification. You can quantify how much protein is present. You can quantify how much amino acid is present using right type of chromatography. Does that make sense? So, the peak area varies depending on the concentration of the analyte. Okay? Please note that I am only showing you the basics of chromatography considering the students here, but just to let you know. But if you need an advanced knowledge, please come and talk to me later. Okay? Now, just to show a very nice example how the chromatogram of a protein look like. It's from the milk, not from the plant. You know, milk you have caseins and beta lactoglobulin and all these things. This is purely based on reverse phase chromatography. Now, based on this, can you tell me among these proteins which one is polar? Which one is polar? It's reverse phase chromatography. Beta lactoglobulin is polar. Kappa casein is polar. Kappa casein is polar, right? It's polar, it comes first. It doesn't like the stationary phase. It has a shorter retention time, right? And this is the most non-polar analyte. So that's how we can separate based on the difference in their polarity, because every molecule will have a difference in polarity, and that is the advantage. That is the thing we leverage here to get the separation. Now let me come to the plant protein world here. You can see how dirty the chromatogram look like. So it's not a nice chromatogram according to me, but still you can separate certain things, and basically the B and C are conglycinin and glycinin peaks in soybean, right? It's soybean, it's soy isolates basically. So you can actually separate them, you can actually identify them and also quantify them. And I can show you more dirtier chromatograms here for plant based one. So it looks really dirty, right? But people try to get more information from this. Please note that it's not the fantastic chromatograph in the world. I hope I have a fantastic chromatogram in the world. This is my chromatogram. So this is something, this is a fantastic chromatogram in the world according to me, because I did this one, or together with my PhD student. So can you see? If you do a really nice chromatogram, it will look like this one, very sharp, nice Gaussian peaks. But proteins will never behave like this. This is amino acid, free amino acid. The proteins are crazy, they interact indifferently, look like so crazy. It's very difficult to integrate this peak and find out the concentration. Even software cannot do it properly. Am I clear? But still we can use them for identification and quantification. What I want to tell here is that you can use reverse phase chromatography to separate proteins by HPLC, but don't think it's an easy go. It's very complicated because separation is always compromised. Okay? This is another nice example of size exclusion chromatography they are using to separate what was that is in pea protein concentrate, and they have legumine, convicillin and vicillin and so on, these things. It's a size exclusion chromatography. Now tell me which one is having the largest size. I have a peak number Z and I have a peak number L. Which one has the largest size? L, do you agree? So in size exclusion chromatography, the bigger one runs first, faster, so it comes out. So this is having the largest molecular weight, and this will be having the smallest molecular weight. And this is actually very complementary to your STS page. Remember my previous class STS page? In the STS page you get the last band here, and that will come here. Okay, the big one will come here. So we often use size exclusion chromatography complementary to STS page. You can also run molecular markers here as a standard to find out the apparent molecular weight in this case also. So we run these two in parallel to see the apparent molecular weight of unknown proteins or known proteins in plant based or alternative proteins. I really recommend this technique in case you want to use for the future. Make sense? Why I recommend? Because plant proteins basically they have a difference in the size usually. For example, usually globulins are very big, albumins are small, so you can use this phenomenon to separate, but you can't separate isoforms, you can't separate there's a tiny difference you can't separate, even in the size exclusion chromatography. Okay? So here is the separation for size exclusion chromatography using separation of rapeseed proteins using size exclusion chromatography. Rapeseed has cruciferin and it has nepin. Cruciferin has 300 kilodalton, nepin is approximately 14 kilodalton. Now tell me which one is cruciferin and which one is nepin here. First one is cruciferin, right? Bigger one comes first, nepin, right? We can also use HPLC to separate amino acids. After hydrolyzing the proteins, you can separate individually, you can identify and quantify the amount of amino acids. You remember in my second or first lessons, I talked about a lot of essential amino acid present, a lot of non-essential amino acids present. All of them were measured by either HPLC or GC. exactly the same technique, right. For example, you have a glycine, we have a methionine somewhere here, tyrosine and so on. So, all these are identified and quantified based on chromatography. The area under the peak is directly proportional to concentration. Okay. All right. So, I have a small group discussion for you. No, I have to skip this one, sorry, at the interest of time. It's okay, don't worry. We will move to the second particular topic of today. I don't teach a lot today, don't worry. I know we are already lagging behind, but I have to cover this topic today. So, in the previous lecture, excuse me. Okay, in the previous picture we had an HPLC with a column, right, and we are pumping the solvent like this one, we are injecting the sample like this, and the separation takes place and things come in this direction, and there is something called a detector here, right, and the detector will detect the ions, and I said you can use UV, you can use fluorescence and so on, and then you get a chromatogram. This is what I explained you past 1 hour, chromatography, right. Now, we can use another type of detector here, that is called MS, right. This is liquid chromatography, can you see? This is liquid chromatography, LC, and then you have an MS detector, and that becomes LCMS. Have you heard about this word? So simple. Please note that in the LCMS, the left hand side is the same as HPLC or UHPLC or UPLC. Now, only the right hand side, what I mean here, the detector is different. Is it clear? Now, I am going to teach you the MS little bit. At the interest of time, I can't go too deep into the MS. Please note that MS is again very complicated thing in the world, I would say. It's a fantastic equipment, but it's a lot to learn. How it operates, what is mean by MS spectra, and how do we understand an MS spectra, and how do we get the information out of MS spectra. It's a little bit complicated. I can't teach you it in half an hour. It is basically a class of a GAN program itself, according to me. It's a lot. But I'll try to cover, considering the class background here, I don't go deeper into the subject. I'll try to give you the basic overview of an MS system and how we can use it for the protein characterization. Is that okay with me, with you? Okay? Yes or no? Yes. Cool. Now, we have been seeing that in the HPLC separation takes place based on the band, and then that reaches to the MS, and then you will get something called MS spectra. Okay? And that is the end product, the MS spectra. In between there is a lot of things happens, right. You have an analyte is coming out of the column, okay. Then the analyte gets you the MS spectra. This is called MS spectra. But to get the MS spectra, there is a lot of things happening in between. I am going to show you exactly what exactly happens here in between to get the end of the day MS spectra, in brief, in short, okay. Now, I need to answer this particular question before going to that particular topic. When and why do we use an LCMS? Can you tell me? Have you ever used LCMS? Yes? Or you must have certainly supplied the sample to somebody to run the LCMS, right? That's often we do often. Why did you supply the sample and why did you run the MS? Okay, you get the separation based on the mass, but please note that that separation and the MS separation is different. The separation of the analyte happens in the column. Remember, the separation of the analyte happens in the column, but mass also a separator. It separates the analytes based on their ions. That I'll come back to you later. You're right, but if you're talking about this separation, it's different. Okay. Any other thing?
It's a derivative of that sample.
It gives a
Derivative of the same sample.
what that information
Means
get?
carotenoids has lot of derivatives.
Okay, you get more information. You
Identify how many derivatives is formed during the reaction.
How do we identify? What extra information does mass spectrometer gives? It gives different peaks. Mass, right? Mass spectrometer gives mass. That is the most important thing we need for the structural elucidation. Do you agree? If I know the mass, exact mass of a compound, 50% of my problem is solved. I am telling you. If I have an unknown compound, if somebody tells exact mass of that one, 50% of my problem is solved. Then I will go to an NMR person and tell you, okay, can you get an NMR spectra? Then I will go to an XRD person and tell you, can you get me XRD? Then I know the structure of the molecule. Am I clear? So, the mass is a fundamental thing we need to get the structure, and structure talks about composition, and the mass spectrometer is a fantastic thing in the world that talks about the structure of the molecule based on the mass. It also gives you the fragmentation pattern, and those fragmentation pattern, for example, this is the fragmentation pattern. This is the mass of the ion. It has a parent for fragment ions here, and this is unique for each molecule, right. In the mass spectrometer, let us say one example, let us say you have a compound that is looking like this one, okay. In the mass spectrometer, it will be fragmented in different possibilities, right, and each fragment will give you one kind of ions here, and it is very unique to that particular molecule. Now I can get you the more information about the molecule, its structure. I get the mass, I also get the fragmentation pattern, and that gets me the structure of the molecule. So, structure elucidation becomes super useful with the mass spectrometry. All right. So, another important advantage is that you can analyze few more topics based on we can analyze a lot of compounds in a very complicated matrix using LC-MS. But LC-MS is super expensive, super expensive compared to HPLC connected to UV detector. I am talking about 10-fold, I am talking about even 100-fold expensive, right. In Indian rupees, we can talk about crores to get a mass spectrometer, a good one, right. It is actually an expensive piece of equipment, but it gives you the fantastic information. All right, these are the basic instrumentation. Since I do not have much time, I will try to take it as a image here only. Is it okay? So, I will show you how the mass spectrometer works. You have a column here and you injected the sample here, right. The analytes are coming out of the column. The one analyte came out, imagine the analyte A came out of the column, right. In the mass spectrometer, we have a chamber called ion source. This is the first important part of a mass spectrometer, and then we have another chamber called mass analyzer, and then we have a last one D, that is detector. These are the three main part of an MS equipment, right. The analyte comes out of the HPLC column. The analyte just comes out of the HPLC column, it enters the ion source, okay. Please note that the analyte that comes out of the column may not be charged, it could be neutral, right. Mass spectrometer can separate ions when the mass spectrometer can separate an analyte when they are ionized, note that point. Mass spectrometer can separate analytes when they are ionized, right. So, we need to first ionize them, right. That is the job of the ion source. So, the analyte A, it is coming and entering the ion source, it will get a charge. The charge could be positive and charge could be negative, depending on what kind of voltage you are applying. It could be positive or it could be negative. Once it is in the charged, then it enters the mass analyzer. Mass analyzer is the one, basically it is a filtering system, right. It filters ions based on their mass by charge ratio, m by z, Okay. It filters the ions based on their mass by charge ratio. I will explain that in later in the next slide. So, that is the job of the mass analyzer. Basically, when the ionization takes place, it's only you do get a lot of different ions. You get A plus, and the daughter of A plus may be A1 plus, A2 plus, A3 plus, so many things will be formed in the ion source. But I don't want everything, I just want A plus, you tell the instrument. Then the mass analyzer will filter only A plus and sends everything out to the waste. That is the job of the mass analyzer. We do not need everything, we need what we want. You can tell to the computer, you tell to the machine, I just want A plus, rest everything should go to waste. Then it will adjust its magnetic and electric field so that it will select the ions based on their mass and charge ratio. And then it will come to the detector, and then you get the so called mass spectra. Make sense? This is mass spectrometry in 2 minutes, it's so much. Now, I will talk little bit about how the ionization happens. Please, I will repeat once again, just keep this in mind. Analytes comes from the column, right. Before you want to get the mass spectra, it has to be ionized. Ionization will be done by the ion source. Okay. The mass filter or the mass analyzer will filter the ions based on their mass by charge ratio, and then the detector will detect those ions. There is a lot of detector, forget about that, that is not important, and then you get the spectra. Is it clear? The detectors are diodes and photo, there are many things. Okay. Now, this is what I was trying to explain. There is a lot of different types of ion sources we have. Okay. The first part, have you heard about different ion sources? Electron impact ionization, chemical ionization, electrospray ionization, atmospheric pressure ionization, atmospheric pressure chemical ionization, atmospheric pressure photoionization, MALDI, okay, MALDI-TOF. There is a lot of different ways you can ionize your proteins or analytes, this part. Okay. This is a very typical unit, a number of different types of units are available commercially. Okay. Depending on what you want to ionize, you can buy the one you want. The most commonly used is EI, electron impact ionization, especially in GC, and ESI, electrospray ionization, especially in the LC, and MALDI, and also we use in the protein science actually. Okay. I will try to cover only these two today, EI and ESI. These are the most common used. And the mass analyzers are many, quadrupole, ion trap, TOF, orbit trap. You must have heard about LC TOF, LC QTOF, LC orbit trap, LC triple quadrupole. There is a lot of mass analyzers are available, and they filter the ions differently. How you want to filter in this particular part, then you get a different piece of equipment. Am I clear? So, depending on the source and depending on the mass analyzer, the price of the instrument also varies. Okay. Mass analyzer is a very important thing, the source is a very important thing, that gives you the high quality information. It is something, if you want high quality data, you need to buy the best mass spectrometer in the world, and that brings you the money, and everything has their own advantages and disadvantages. Is it okay? Now, I will talk little bit about ionization, especially electron impact ionization. Okay. Now, the analyte is coming here from the column, right, and this is an analyte, right, and this is entering the ion source now. It came inside the ion source. We apply a high voltage electron at high energy, right, and this electron hits the analyte, right. After hitting the analyte, it will knock off the electron from the analyte. When you knock off the electron from the analyte, you get analyte, radical, anion, sorry, cation. Okay. Now, can you see you got a charge on the analyte now? Originally it was neutral, now you got a charge. Okay. But the energy that electron hits is so fast that the molecule can be fragmented. Okay. So, you not only A plus you will get, you will also get the fragments of A plus. I will write it as A1 plus, A2 plus, A3 plus, so on. Its fragments are basically, let's say, this is an analyte. It can be fragmented everywhere, okay, because of the impact of the electron. So basically, you get a different types of analytes, right? And these analytes are now pushed in the vacuum in a chamber like this one, okay. These analytes come to the vacuum chamber like, and here we apply the magnetic field. We apply the magnetic field. Are we running out of time? I need some 5-10 minutes at least. Okay, okay. We apply a magnetic field, right? We apply the magnetic field. Now you know that ions can be deflected based on the magnetic field. Ions will move in a different direction. They go crazy when you apply a magnetic field. All right. Now we have ions coming, which is A plus and A1 plus and let us say A2 plus. Now A1 plus is a bigger size, A2 is second big, and then your smallest one is A3, imagine like this, right? And they deflect under the magnetic field in a different ratios because they have a different mass by charge ratio, okay. Usually the smallest one deflects much easier than the biggest one, right. So basically the smallest one is A2, it will deflect more compared to A plus, the bigger one, right. Now what we have is actually the spatial separation of the ions based on their mass and charge ratio, okay. In the small, this is all happening in fraction of second, fraction of second basically, it's happening so fast, right. So they separate in the mass analyzer in the here, and then they will be detected as an individual ions because they are separated here based on the mass by charge ratio. Is that clear? And this is the basic principle of the mass spectrometry, the ionization and separation. So you have a high energy electron, it hits the analyte, then the analyte can get a charge, but it not only get the charge, sometimes it will also break the analyte into multiple pieces, then you get a bigger fragment, smaller fragment and so on, and all the fragments will enter the mass analyzer here, and under the vacuum, under the high magnetic field strength, they will separate spatially based on their mass by charge ratio, and at the end of the day you can detect them as a MS spectra. Is it clear? Are you getting my point? Okay, okay. Yeah. Now just to show one example, you have phenylalanine, this is an amino acid, it can be fragmented into different pieces, can you see? And this can move in a different velocity if you have a charge, and this can move in a different velocity, and this can move in a different velocity in the mass analyzer, and that can be used as a separation principle. You get a mass spectra something like this for each fragment, okay. This is mass. And then I will briefly go through the electrospray ionization. This is called ESI, often used in the LCMS. It is a very similar to the previous one, EI. EI is the electron impact ionization. Now I am talking about the electrospray ionization. It is often used in the LCMS, liquid chromatography, not in the gas chromatography. So what do we do here is that the analyte is coming again here from the column. This is my analyte, okay, and the solvent will have this solvent charge and so on. Basically we spray, I hope that I have a figure, that one, yeah. We spray the analyte in a small nozzle. You know the scent bottle, you spray, it becomes an aerosol. Exactly that thing we do here, okay. With some gas we spray the analyte, it becomes an aerosol, right. And the temperature will be here in maybe 200 degrees Celsius, maybe 300 degrees Celsius, okay. And then it will get kind of a small droplets of the ions like this, right. At certain point the droplets will be broken, then you will get the individual ions with charge, right, individual ions with charge, and that will go to the mass analyzer. Very simple procedure, but it has a lot of complicated machinery to do this one. So basically you have an ion, in the liquid chromatography you have a liquid, okay. The liquid is mixed with gas and you should actually spray them, and then there will be formation of the bubbles with charge. I do not go in detail about all these things, do not worry. Just remember that you get the ions with the charge. In ESI source, you get a positive charge by adding a proton to the molecule. This proton can come from the solvent. You can also get, you can remove the proton from the molecule, then it is called negative mode ionization. Have you heard about ESI positive mode ionization, ESI negative mode ionization? Now, that is something related to the structure of the molecule. If you have an amino group, it can easily protonated, so we run it in the positive mode, M plus H. Now, you get the charge. Basically, it is the molecule plus a mass of the proton. The mass of the proton is 1. Okay. So, imagine the molecule has some, in this case the molecule has 721 as a molecular weight, and it added 1 proton, it becomes, sorry, 722, and you get 723. Okay. And in the negative mode of ionization, you remove a proton from the side chain and it gets the charge. And the charged ions will again move in the quadrupole and so on, and they will get separated. Okay. I just have to skip this one. Now, I will just, there is a lot of different types of mass analyzer. Unfortunately, I can't cover it today at the interest of time, but I will just give you how we are using this mass spectrometry in protein analysis. Okay. So, we do the ionization of the proteins, right, and they are charged now, you can see different ions, and they are separated in the mass spectrometer, and you get individual chromatogram like this, and each chromatogram, it may be the peaks is associated with the protein, different type of protein, protein 1, protein 2 and protein 3, right, and under each peak you have a mass spectra, and that will help us to identify the unknown proteins, because the mass of the protein is very unique, right. That will help us to identify. Not only identification, we can also use it for the quantification. Why? Because the area under the curve of each mass fragment is directly proportional to concentration. So, this is called intact protein analysis, intact protein analysis. We also call it as top down proteomics. So, basically the analytes are ionized and supplied to the mass spectrometer, and you get a peak and a chromatogram and a spectra. You can use it for the quantification or also identification. Then we have a peptidomics, or what we call bottom up approach. In this case, what we do, we do not inject protein directly to the mass spectrometer. Before injecting the protein, we hydrolyze the protein into individual peptides. Then the size of the protein is shrunken, right. Then you get a peptide fingerprint, and that will help us to understand which amino acid is hitting one another, because each peptide will have its own fragmentation pattern, and we get another information here. By combining these two information, we can talk about the structure of the protein, the molecular weight of the protein, the sequence of the protein, and these are the two approaches we are using to do protein characterization in food science. I only took it only half an hour to explain mass spectrometry. As I said, it is a complicated field, and I really need another 6 hours to explain this in detail for you, but that is not necessary considering the background of the classroom. That is not me. Okay. Okay, we are coming towards the end of the lecture. So, I just wanted to mention about mass spectrometer. Just remember that the mass spectrometer is nothing but the instrument that measures the mass by charge ratio, and that is an important tool to identify and as well as quantify proteins, amino acids, whatnot. It can be used for almost everything. As long as the molecule ionizes, we can use it in the mass spectrometer, unless they are extremely big. Okay. Principle is simple. There is an ion source where the analytes will be ionized. Only ions can travel in the vacuum under magnetic and electric field. Okay. And there is a mass analyzer next to it, and it will filter the ions based on the mass by charge, and then we will get a detector that will detect it as a mass spectra. That is the basic principle of mass spectrometry. Okay, that is it for today. Maybe today, just to conclude, we have studied too many things. I can say that today we covered about spectroscopy. Can you mention which one we covered to begin with? We started with primary structure of a protein, Edmonds degradation, secondary structure of protein with FTIR, tertiary structure with fluorescence and also with XRD, and then STS page for the separation and molecular weight determination. Then we learned a lot about chromatography, the basic principle of chromatography, polar, polar and so on, TLC, column chromatography, size exclusion chromatography, reverse phase chromatography, normal phase chromatography, affinity chromatography, and then we came to mass spectrometry. When you get a slides, just go through the slides once again. If there is a question, get back to me. With that, thank you so much. And that's it for today, and let's see tomorrow.